Good morning, good afternoon, and good evening. My name is Elena Moreno. I'm the Senior Project Manager for the Global HIV Vaccine Enterprise at the International AIDS Society. And it's my pleasure to open this meeting on accelerating early phase HIV vaccine clinical research. This event marks our annual stakeholder meeting where we convene HIV vaccine and R&D stakeholders with the aim to share perspective on priorities and factors to consider in advancing vaccine product development that encompasses design, clinical testing, and funding. I would like to welcome all participants as well as our chair, speakers, panelists, rapporteurs, and facilitators that are with us today. As you can see on the agenda, we will have 3 presentations followed by a panel discussion. We will later split in 3 breakout rooms where we will have more focal conversations. We will have a break, and after that, we will summarize having a report session with a guided general discussion. And we will finalize the event with a second panel discussion with questions and answers at the end. You can, at any time, even we have that session at the end, you can ask questions through the questions and answer box, please. You see that you are all muted, but you can write that at the end on the box. So it's now my pleasure to introduce the Vaccine Enterprise Advisory Chair, that is Susan Buchbinder. Susan is the Director of Bridge HIV and Grant Funded HIV Prevention Research Unit, housed in the San Francisco Department of Public Health, and a clinical professor of medicine, epidemiology, and biostatistics at the University of California, San Francisco. Susan, the floor is yours. Thank you.
Thank you so much, Elena. So this is an exciting time in HIV vaccine research. We have these new tools like mRNA that can accelerate our ability to iterate, and we have a robust scientific agenda with hypotheses about how best to create vaccines that will generate broadly neutralizing antibodies, as well as T-cell based vaccines. However, we're facing some operational, regulatory, clinical, and funding challenges that can slow the pace of HIV vaccine research. So the Vaccine Enterprise is convening the stakeholders meeting to identify and address some of these challenges and to help chart a path forward to accelerating our experimental medicine portfolio of studies. Today's audience is broad and diverse, and we hope the format will facilitate a productive discussion on how to accelerate HIV vaccine research, how to ensure that the most promising approaches move forward swiftly, and to identify and address the biggest challenges we face in achieving these goals. Elena went over the structure of today's meeting. You can see that it's really been structured to maximize participant involvement and robust discussion. And with that, I want to thank you so much for joining today, and we look forward to the rich discussions. So we'll start with our first 3 presentations. The first one is by Kundai Chienze from IAVI Kenya, who's going to be talking about an overview of the pipeline of products ready to enter or already in early phase clinical testing. Kundai, thank you.
Thank you, Susan, for that kind introduction. And so allow me to just fix the display. So I hope you can all see the slides clearly on your end.
Perfect.
Thank you. So I'm going to move very quickly into the presentation to save on time, and acknowledging the donors that make the work that we do within IAVI possible. However, in today's talk, I will not be focusing on the work that IAVI does, really focusing on the work that the broader field is accomplishing over time. Sorry, I have a bit of a taskbar that I just need to move out of the way. Okay. All right. So it's important for us to remember the state of the epidemic as we stand in 2022. So 84.2 million people have been infected with HIV since the beginning of the epidemic. And there has been a lot of progress that has been made in the treatment and prevention space, leading to a reduction in new HIV infections over time since the beginning. However, in 2021, we still saw for 1.5 million new infections happening across the globe. And so this really highlights how HIV continues to be a significant global challenge. And I know many of you will agree that a safe and effective HIV vaccine is still needed to achieve a sustained end to the epidemic. So several efficacy trials have been conducted thus far since 2003. And the one efficacy trial, the RV144, did show modest protection. However, since then, there are few trials that have been done as a follow-up to that trial, and they have not been able to replicate these results. The AMP trial that was conducted, where we saw results being presented in previous years, I believe it was in 2020, we did see protection that was, whilst overall the trial did not show protective efficacy, there was protection observed with VRC01 against sensitive strains. And this result does give us proof of concept that potentially it is possible to protect from HIV infection by the delivery of broadly neutralizing antibodies. However, it did sort of highlight the need to use a combination of antibodies, and also suggested that high titers might be required to achieve protection. So important lessons have been learned from these efficacy trials. However, the broader field has realized the need to continue to identify new immunogens and approaches if we are to realize the goal towards an HIV vaccine. There are important scientific questions that need to be answered as we move forward. And of course, the key thing I think Susan already alluded to about the approach to induce broadly neutralizing antibodies through vaccination. Can we really do this? It has not been done before in humans. And if we can do this, how can we also induce multiple BNAbs against that target the different epitopes on the HIV envelope? And how can you actually do this in a single individual, for example? So what kind of T-cell responses would be needed to offer protection? Some of the efficacy trials that have been conducted so far were actually looking at T-cell based vaccines, and those were not successful. So going forward, which kind of responses do we actually need to elicit to be able to achieve protection? And if we do now sort of see results on both the BNAb and the T-cell space, how do we effectively combine both antibody and T-cell approaches? And there's also a question that has come up if, T-cells, whether the use of T-cell vaccines can actually reduce the level of neutralizing antibodies that are required to offer protection. And there are also many questions around the delivery of epitopes, how many doses will ultimately be needed, and the use of technology to assist us to deliver the needed. In this era of experimental medicine trials, how do we accelerate the process of iteration to arrive at proof of concepts and determine the actual vaccine candidates and regimens that we would actually be taking forward towards licensure? So the current vaccine approaches that the field is focusing on is around the generation of broadly neutralizing antibodies and of course a T-cell based vaccine approaches. There are others and I will not dwell on those too much, but the field very broadly is also focusing on the use of adjuvants, looking at HIV epitope expression systems, for example, whether to deliver by protein, nucleic acids, and there's a lot of discussion around mRNA platforms. There's a lot of work that's also looking at dosing and boosting, whether fractionated dosing, for example, is relevant for certain vaccine approaches. So we'll start with focusing on the BNAb-guided vaccines. So ultimately here, the goal is to develop a vaccine that will elicit sustained protective levels of BNAbs in humans. And if you look to your right on the screen, you will see here the envelope, the HIV virus in the envelope in the areas where BNAbs actually bind. And you will see that the BNAbs that have been isolated thus far can bind to multiple sites on that HIV envelope. So the hypothesis really is to ensure that we have coverage against the huge diversity of global isolates. The hypothesis is that an HIV vaccine, for it to be effective, it will need to consistently induce BNAbs against multiple sites on the HIV envelope in most vaccine recipients. So this is just a look at the approaches that are being looked at in terms of generating BNABs. And in general, the principle here is, you know, in an individual who is HIV uninfected, where they have BNAB precursor cells, or what we call naive precursor cells, that these can be matured, firstly to engage with them, and then begin to boost them and drive them to mature, to become and have the actual characteristics of broadly neutralizing antibodies. And you will see, and the thinking here is that for us to actually be able to arrive at BNABs, you will require a series of boosts before you have broadly neutralizing antibodies that are well matured and broad. And so there are several approaches that are several groups that are looking either at lineage-based vaccine design, germline targeting vaccine design, or immunofocusing vaccine design, with several examples that you will see on the right there. So when I, looked at the work that's currently ongoing in this space, and a lot of experimental medicine trials that are currently being done, there is a very rich pipeline of trials that is looking to generate broadly neutralizing antibodies. And this just to give you a brief snapshot of what is currently ongoing. And that, so for example, the IRVG001 trial, which completed, was the first trial that really showed a proof of concept around the germline targeting approach, that it is actually possible to engage the naive precursors. And this was using a protein trimer. So this trial actually did not go into including the boost. It was really the first stage. And this is really probably the first of many things that need to be done subsequently. And so as a follow-up to this G001 trial, and results have been shared in various meetings in the past, there are two other trials that are currently ongoing, the IRVG002 and G003 trial, that are also looking at the EOD GT86 dimer that was delivered in G001. The difference here is that these two trials are looking at the same sort of immunogen being delivered via mRNA. And we will be talking about, about how mRNA is beginning to change the field, and how we are looking at conducting multiple trials in the coming years. At the same time, there are many other trials that are being conducted through the HVTN that are also looking at generating BNABs, and some of them are targeting different BNAB epitopes. In the next sort of 12 to 24 months, there are many trials that we are looking to have begin. And you will see here just a snapshot of 9 that will be beginning in the next 6 months, estimated to begin in the next 6 months or the next year. But I wasn't able to show all the over 16 other trials in addition to this. So this comes up to about 25 new experimental medicine trials that are expected across the globe, where they're looking at the US, Europe, and Africa. And again, what you then see from all these trials is they are also looking at targeting different BNAB epitopes over time. In terms of looking at vaccine strategies that are geared towards the generation of T-cell responses, there are a few that are worth highlighting. And the first one here is really looking at one approach that is focusing on conserved viral epitopes, and one approach that is being pursued through the University of Oxford with the Tom Hankey group, just looking at functionally conserved regions of HIV proteins, and really focusing on those that, if there is a mutation, this would sort of render the virus, would sort of have a cost on replication fitness. And so this is targeting regions that are common to most global HIV variants. There's another approach that is being led by Gaurav Gaye that's looking at networked viral epitopes. and really leveraging structure-based network analysis. And this has identified the regions of highest importance to HIV protein, sort of tertiary and quaternary structures. And you have CD8 T cells targeting highly networked regions, and this has been associated with control of HIV natural infection, for example. And so also developing vaccine approaches that would be that would be generating T cell responses. Another exciting approach in the T cell space is looking at CMV-based human vaccines. And really, this is a replicating vector to safely mimic natural infection and eliciting responses. And really here focusing on generating sort of unconventional or non-classical MHC E-restricted CD8 T cell responses. And again, just to give you a snapshot of some of the trials that are ongoing in the T cell space, we have the HIVCO-06 trial, which is really looking at these conserved immunogens that are being delivered either via ChIMPAD vector or MVA vector by the University of Oxford, and then CMV vectored vaccine in the VIR-1 1111 trial led by Louis Picker, and that's also currently ongoing, and the results of this trial have been shared in some fora. But because this is a prototype vaccine, there are subsequent trials that will be starting later in a few coming months. For example, the VIR-1388 that will be rolling out soon, starting in the US. There are also some conserved follow-up trials to the conserved immunogen sort of approach for the T cell space being delivered via mRNA. There are several other vaccine approaches that are also ongoing that are looking at other virus vectors. For example, the AD4, apologies there, I think that the protocol trial there is a mistake. And then there are several other trials that have been done, for example, Robin Shattuck and his group that have been looking at other native trimers, mosaic trimers as well, some SOSIPs, CONEM, and looking at mosaic SOSIPs as well. And all those products sort of being delivered with an adjuvant MPLA, and those trials are ongoing in Europe as well. I do want to highlight a few trials, the last two there, the RV460 that's being conducted by the MHRP, and one by HVTN 137 that's a trial led by Julie McElrath. The reason I'm pointing out these two trials is the focus where they are looking at different adjuvants and seeing and comparing different adjuvants in the different trials. And so in the next sort of slide, I just want to touch a bit on the work around the adjuvants that's being done and sort of the rationale behind this. So adjuvants have been effectively used to improve immunogenicity of recombinant vaccines, as well as increase breadth of protection. Very useful, have also been used in the past in terms of sort of being for dose sparing or reducing the number of doses, which can be very useful in pandemic or epidemic responses. They can also be used to sort of generate long-term antigen-specific plasma cells and memory B and T cells. There are multiple studies that are evaluating various adjuvants, as you saw from the previous tables that I showed you, and also the last two that are highlighted. And a key question in all these adjuvants sort of comparing studies is, we will be able to understand and learn which adjuvants work better with certain vaccine approaches, and be able to understand how to smartly use them to enhance certain properties. For example, if say in the germline targeting, responses in the germinal center are very important, and there are some adjuvants that sort of do improve germinal center responses. And so really being able to understand, one, how they work, and being able to pair them smartly with the various immunogens or the vaccine approach that is being leveraged. So in closing, there have been disappointments in recent efficacy trials, but there is a range of new exciting vaccine approaches that are in early development that are based on rational vaccine design. There is a rich pipeline of products in experimental trials and early phase 1 trials that are evaluating BNAB generating approaches, T-cell approaches. There's a range of adjuvants that are being evaluated and will inform smarter use and pairing across vaccine approaches. Also looking at various epitope expression systems, looking at mRNA, comparing that to protein delivery, also looking at dosing, really trying to understand, for example, whether fractionated dosing is better in some cases or not. We didn't really talk about mRNA, but just to highlight that it is offering opportunities for accelerated evaluation to allow for rapid iteration towards candidate vaccine regimens. One thing I didn't highlight in the presentation is I was looking at all the incredible, the number of trials that are being done globally. It was very clear, whether looking on clinicaltrials.gov or PACTA, the WHO sort of database, that a lot of these trials are being conducted either mainly in the US. There are several that are being done in Europe, very few being done on the other continents, for example, whether it's Africa or other parts of Asia. So it is important to begin to also begin to talk about how we begin to do these trials across the globe and increase our ability to answer important questions quickly and contribute to the answers. And so I want to thank everyone and acknowledge the whole field of researchers who are tirelessly pursuing in HIV vaccines, all the trial participants that volunteer in clinical trials, and the funders who make the work possible. And of course, very importantly, the communities who partner with us in this work and continue to challenge us to do better. Thank you.
Kundai, thank you so much for that fabulous overview of the pipeline. And we'll move next to Bart Haynes, who's going to talk about promises and challenges of new early stage clinical testing strategies. Bart is obviously from Duke University.
Thank you so much, Susan, and thank you so much, Kundai, for that great first talk. So with regard to some select strategies that are chosen to highlight in this brief talk that are promising and offer some hope for making a broadly neutralizing antibody vaccine, and then we'll go into some of the challenges. So just as a conflict of interest, the Duke CHAV-D team, and including myself, has some patents submitted for some of the concepts talked about in this talk. So what the field is working on, as Kundai said, is the induction of high-titer durable polyclonal serum and mucosal BNAB responses to multiple HIV envelope epitopes that will protect against HIV transmission. And the goal for the field over the next 4 years at least is proof of concept that a BNAB vaccine can be made with the criteria to induce at least one type of broadly neutralizing antibody to achieve greater than or equal 50% serum breadth against R5 tier 2 transmitted founder viruses with antibodies that bind to a BNAB epitope in monkeys or humans. And secondly, to induce durable serum and mucosal broadly neutralizing antibody levels for a minimum of a year in monkeys or humans. So let's go directly to what some promising strategies are available. And the first is modified mRNA lipid nanoparticle technology. Our CHAV-D first got into this in 2016 when we brought Drew Weissman into our CHAV-D. In 2017, because Drew didn't have an mRNA for HIV at the time, but we wanted to rapidly see if this translated from mice to primates. We used what he did have, which was a Zika vaccine at the time against PRME. We were going to use it for 4 immunizations. After 1 immunization, the titers were so high, we went ahead and challenged all the monkeys with Zika, and all 100% were protected. And this was published, and this paper recently was awarded by the Portuguese government for this award, BIAL award. That's Drew Weissman there accepting the award, because this was the first manuscript published that used a nucleoside modified mRNA lipid nanoparticle platform. In NR-CHOP-D, we've gone on and shown in this paper that modified mRNAs with Drew Weissman can encode for trimers, complex scaffold nanoparticles, in this case, ferritin nanoparticles, and also cell surface GP160s. We've set up a strategy of mRNA design that from design to release, we hope will take around 10 months. We're not down to that quite yet, but I think that's much more than a reasonable goal and maybe even shorter. And the design then goes to the Weissman lab, comes back to the CHOP-D for in vitro screening and expression, then goes into mice to make sure that it expresses well in vivo. This is very important. We've had constructs that have made it to engineering runs, but that did not express in vivo well. So we're doing those screens very early. If it fails in vivo expression, it goes back to design. If it passes in vivo expression in mice at small doses, such as 1 microgram immunization doses, goes to mRNA manufacturing and to fill finish. So what are the advantages of this platform? And Kundai's mentioned some of these. Number one, we believe in contrast to COVID that these will be giving durable antibody responses, and Norbert Party's published this, Kevin Saunders has published this. Secondly, that it can induce high levels of T helper cell responses for antibody production, again from the Weissman group. And it's very practical for multiple component vaccines, and it appears to be much more cost effective than protein vaccines. The short production time for mRNAs compared to proteins is essential for iterations, and also much faster than for production of live or inactivated viruses. Therefore, we can test many vaccine candidates much faster, and that's something we're going to have to really do much quicker than we're doing now. But it should be mentioned that the mRNA platform is not going to be a panacea, because the key issue for HIV vaccine development remains immunogen design. So another promising aspect is the concept of germline targeting, and this is an illustration that Dennis Burton and John Mascola and I published a couple of years ago to explain sort of what generally the field is doing with targeting immunity, because these lineages, as listed here below, are the lineages that Kundai mentioned that we're trying to induce antibodies against. Because the lineages are disfavored by a variety of reasons, we're having to stimulate the precursor B cells and then use sequential, slightly different immunogens to expand and the lineages to get them to be selected for B cells that are affinity matured and have the characteristics of broadly neutralizing antibodies. In effect, we're having to engineer the immune system to do what it fundamentally doesn't want to do particularly well. And so with some progress in germline targeting, Bill Schief and IAVI G001 has a protein EOD-GT8, again, as Kundai mentioned, that induced B cell antibodies, B cells making antibodies with characteristics of BRCA1 class CD4 binding site, BNAbs. And IAVI G002, Bill's using mRNAs that encode for EOD-GT8, and these have also induced B cells with BRCA1 precursor characteristics. Bill also has HV10-302 with mRNAs that encode for transmembrane-bound HIV envelope, BG505, a clade A envelope that has induced autologous neutralizing antibodies, as reported by David Montefiore a couple of weeks ago in the HV10 meetings. Rohir Sanders, this is a slide from Rohir Sanders, and Rohir has germline targeting immunogens for V2 and CD4 binding site on the same immunogen. And this is the AVI C101 trial. And in preliminary data just presented at the CAVD meetings, GT1.1 is the immunon. It's immunogenic in humans and induces BRCA1 class responses in a subset of individuals. Quite impressive data showing a cryo-EM structure at that meeting that shows that the antibodies induced have the same signature structure as the BRCA1 antibodies themselves. Then moving to Julie McElrath's trial, Dennis Burton with the BG505 SOSIP trimer designed by Rohir Sanders and John Moore have been in a number of adjuvants and have induced tier 2 autologous neutralizing antibodies. One of our trimers, CH505, has done the same thing with the adjuvant 3MO52 in the HV10 300 trial. And Leo Stamatotis has an upcoming germline targeting for CD4 binding site B-cell precursors, and he's had success in germline targeting and Fredolf's broadly neutralizing antibody germline knock-in mice. Now, have any of these trials induced broadly neutralizing antibodies to completion? Well, recently we reported at the CAVD meetings last week and into HV10 meetings a couple of weeks before that, the trial HV10-133 run by the HV10 with Lindsay Baden, Steve Walsh, and Nathan Erdman of an Emperor GP41 membrane proximal external region, or Emperor peptide liposome that we had developed in the CHAVI ID several years ago. The trial was 20 vaccinees. Interestingly, the trial was stopped after 2 immunizations in 20 and 3 immunizations in 5 vaccinees, after one of the vaccinees developed a likely PEG-associated, polyethylene glycol-associated anaphylaxis event. Fortunately, that individual rapidly recovered. And we went ahead and studied the trial. And even though after only two immunizations, 95% serum response rate against the Emperor epitope was seen, and 85% very reasonably robust CD4 T-cell responses were seen. Interestingly, when we profile the repertoire, we found that this vaccine-induced tier 2 heterologous neutralizing antibody B-cell lineages that are polyclonal, and that there are over 30 different lineages were isolated, and they use 4 different V heavy chains. And so, number one, it induced a polyclonal B-cell response. And secondly, the breadth of the immunogen that the immunogen induced was primarily clade B focused, as has seen with the prototype immunogen against this epitope 2F5, but hit 40% of tier 2 isolates from data from Mike Siemens' lab. But most importantly for us is that for B lineage design, where this immunogen was designed for the unmutated ancestor of the prototype Emperor antibody called 2F5, the B-cell lineage design on one BNAB B-cell lineage could lead to multiple BNAB B-cell lineages induced. It's very encouraging. And on the right there is the crystal structure from Adi Ofek showing the epitope of the linear epitope in yellow with the heavy and light chains of the BNAB. And so what we were able to observe for the first time in humans is the isolation of unmutated ancestors, as well as early intermediates, late intermediates, as well as lineages that went on to full broadly neutralizing heterologous breadth, with gradual acquisition of ability to induce neutralization against easy to neutralize isolates, and then more affinity matured antibodies that can neutralize the more difficult isolates. So the next steps here to remake this immunogen without polyethylene glycol, and that's been done, and to boost, can go back and boost to increase serum titer and antibody breadth to get higher levels of both breadth and neutralization. So what are the challenges that the field faces right now? Well, the first is that the envelope is heavily coated with glycans that mask these epitopes. As you can see, the blue molecules moving all over the place are the glycans on the yellow polypeptide backbone, intermittently covering the broad neutralizing epitopes that we're trying to induce. And so the solution here is that we need to induce high affinity binding immunogens that can bind or accommodate these glycans. So a lot of work is going on designing immunogens that can bind directly to the glycan shield and or accommodate these to get to these polypeptide backbones. The second is immune tolerance, and that, as I said, the broadly neutralizing antibodies are disfavored, totally different than for COVID, where the body is delighted to make the RBD neutralizing antibodies. Here they're disfavored by immune system, leading to autoreactive or unusual BNAbs that need to have traits of autoreactivity or unusual aspects of the BNAbs, that leading to the fact that these are rarely made. And so the solution here is to use, and we and others have published this, is to use in the knock-in mice models, to use strong adjuvants to transiently overcome the tolerance controls in a safe manner. The third is a recent finding of roadblocks called improbable mutations. The broad neutralizing antibodies have high levels of changes in nucleotides or mutations, and many of these critical changes are required for BNAb breadth to develop and for potent antibodies to arise. But these rarely occur. And so the solution here is to design boosting immunogens specifically to bind and select B-cell receptors with these rare mutations. Next is that easily made or antibodies are frequently off track. They go away from making broadly neutralizing antibodies. And these are not broadly neutralizing antibodies, but they can divert the immune system in the germinal centers. And so non-neutralizing epitopes on the HIV envelope can induce antibodies that outcompete these rare broadly neutralizing antibody precursors. And secondly, they can also bind to the same broadly neutralizing epitope and block BNAb activity. And we're seeing this in in one of our human clinical trials. And so the solution here is to design boosting immunogens to induce very high levels of high affinity broadly neutralizing antibodies that can break through and overcome these roadblocks. And then Kundai mentioned the AMP trial, and that has raised a concern that the levels of broadly neutralizing antibodies that are going to be needed to prevent HIV infection appear to be relatively high and are going to have to be of long duration. And of course, this trial studied the infusion of the VRC01 CD4 binding site BNAb. Overall, there was no protection, but for susceptible isolates, it was protective. And the serum ID50 for those isolates needed to achieve 80% protection from transmission of susceptible strains was a titer of around 200. And so another huge challenge is we've got to move faster. We need to have more GMP manufacturing capacity in the field. At Duke, for the Duke CHOP-D, we built a manufacturing facility just for this purpose. And that includes fill finish capabilities so that we can do everything on site for both proteins and mRNA immunogens. So in summary, the field has made progress on inducing proximal EMPER and CD4 mimic CD4 binding site BNAb precursors by germline targeting. The first induction of full BNAb development for GP41 EMPER B cell lineages. We've now obtained this and seen in humans the spectrum of going from germline to heterologous neutralization. So it is possible. And though the goal is now to expand what we've learned with these rules that we've learned of how to design primes and immunogens and go ahead and induce CD4 binding site, V2 glycan, which is another site on the envelope, V3 glycan, another BNAb target, and EMPER antibodies that are more potent and more broad with a multivalent immunogens in humans. And also to add T-cell vaccines to a successful BNAb vaccines to perhaps lower the amount of BNAb needed. We really are so fortunate to have such steadfast funding from a number of our funders, including the NIH, Gates Foundation, lots of collaborations, and with our partner clinical trials, the HVTN. A lot of folks, it takes a village to do the work that I've just talked about regarding from our CHOP-D, and the clinical trials team is just amazing. I'll stop there.
Bart, thank you. That was a fabulous talk. Very insightful. We're going to turn next to Maureen Luba from Cooper Smith in Malawi, who's going to talk about a community perspective on why we need an HIV vaccine, given that we've got long-acting PrEP on the docket.
Just checking that you can hear me and we can see my slides in presentation mode.
Perfect.
Yeah, thank you so much. And thanks a lot for having me this afternoon. As already introduced, I'm Maureen Luba from Malawi, working with Cooper Smith. And this afternoon, I've been asked to share, moving away, slightly moving away from the science, to share a community perspective as to why we still need an HIV vaccine when actually a long-acting PrEP is just around the corner. And before I go into my presentation, let me just start with a disclaimer that the views and the opinions that I'm going to share on this call are my own and not those of the organizations that I work from. Okay. Okay, for some reason I can't move my slides. I don't know, am I slide? Okay, are my slides moving?
Yes.
Okay, perfect. Yeah. Yeah. So this is the outline of my presentation. I know I only have 10 minutes, so I will not go into details about that. But I just wanted, I guess, to quickly start with a reflection of the history of vaccination. And I think this is important, especially in responding to the question, why do we still need an HIV vaccine when we still have other options, long-acting options that are available, some that are in the pipeline and soon to be available on the market. I think for those that have followed the history of vaccination, I think you agree with me that vaccination or immunization has such a long history from as early as the 15th century, when people would intentionally, expose themselves to viruses, to diseases as a way of preventing themselves from illnesses, a process which was actually called variolation. And I think if you dig deep actually into that history, you find that, such practices, were being practiced from as early as, 200, BCE. And since then, such practices were expanded, modified, and out of that, there have been a number of vaccines that have been developed for different diseases, conditions such as polio, smallpox, rabies, diphtheria, influenza, yellow fever, monkeypox, HPV vaccine. The list is actually endless. And of course, the WHO and many other stakeholders actually do acknowledge that, vaccines have saved more human lives than any other medical intervention in the history. So it's really important to understand the history and why do we still talk about the vaccine, and especially for HIV, which has been with us for so many decades now. And we've just learned from Kundai and Barton how challenging and complex it has been for us, to find, this HIV vaccine, given the nature of the virus that we are dealing with. And again, just, reflecting on the current landscape, HIV landscape, we know that there have been moments when we've celebrated these different scientific breakthroughs in HIV prevention. And I think I'm glad to say that now, as you all know, we have quite a number of HIV prevention options that are available on the market that can be accessed, by different populations. Yet on the other side, you see that the new HIV infections, and I think I also mentioned this, she gave us like an epidemiological picture of the HIV, that the new HIV infections are not falling fast enough as we would want them to. And the UNAIDS report is just a reminder. Of course, we know there's been COVID at play. But it has reminded us, that the HIV response is still in danger. And there's a lot that needs to be done for us, to achieve the 370 new HIV infection targets for 2025. And I think it's also important for us just to reflect on some of the lessons that we have learned from decades of HIV prevention programming and implementation. This is what have been some of the challenges, more broadly in terms of HIV prevention, implementation. And I think one key thing that we've actually learned from implementing different HIV prevention services, but also interventions, is that addressing barriers to access is very key. I think one thing that we have learned is that, availability is not always equal to accessibility. You may have HIV prevention tools, available at a clinic somewhere, but those, because of, different structural barriers, people may not be able to access those interventions or services. So we need to make sure that we remove those barriers. Decades of HIV prevention programming have also taught us that people, need options that respond to their needs. People are not homogeneous. And sometimes I think we get caught up into, putting people, grouping people into different population groups, HIV, key populations, women. But when you look at HIV, for instance, right, if you take an HIV from, Kauma, which is a semi-urban location in Malawi, to a girl from Talile, which is a rural, location in Malawi, their needs are different because of the social, economic, and demographic variables that do impact the decisions and the risk that they meet in their daily life. So it's important, to even, consider those differences, designing different HIV prevention intervention. That's even important as well as we are thinking about the different designs of HIV vaccine that we would want to produce, and how would it respond to all those different needs of populations that we have to deliver to. The point of delivery, how and where someone access the intervention really matters. I think we have seen that most of the interventions have been pretty much centered around the health facility, right? But of course, with COVID, I know it actually pushed us, powered some innovation, for us to be able to differentiate, to move out of the facility setting and deliver the services into the communities, reaching people where they are, right? And this is something, the trend, that, we need to adopt moving forward. We need to differentiate, making sure that we are reaching people where they are, bringing services closer to the people. Cost matters, right? We need to make sure that, one thing that we have learned from HIV prevention programming and implementation is that, it's important, to make sure that HIV prevention interventions, cost effective, affordable, and people should be able to, to access them, right? Affordability, it impacts, accessibility as well. Adherence, I think, remains a challenge, right? And I think that's why we've been talking about long-acting HIV prevention interventions, and I think with a number of HIV prevention options that we have, the biggest challenge has been adherence, because of the frequency or the demand, or the frequency of one has to either visit a facility or take that HIV prevention intervention, right? We have also learned, especially for women, that, do face a lot of challenges because of their socioeconomic status. They do prefer district HIV prevention options, and choice is important, right? So again, going back to the question of why do we still need a vaccine when we do have quite a number of HIV prevention options available and accessible, and some in the pipeline, right? And I just wanted again to reflect on these two important codes, one from IAVI and another one, of course, from Dr. Fauci, which again acknowledge and emphasize on the fact that we can't eradicate, HIV without a vaccine. We do need an HIV vaccine, and the no questions. It's a must. And I guess that's why we're having this conversation here today, to try to figure out how can we address the challenges that we are facing. We know that an HIV vaccine could provide a long-lasting protection, which is what we want compared to other HIV prevention options that are currently available, and it could be given to broadly to a wide range of population groups, wide range of people, and give us the immunity that we want. An HIV vaccine would also likely going to be cost effective when compared to other HIV prevention options that we have are currently available. But also, if you consider, someone getting infected and being initiated on treatment and being on lifetime treatment, when you compare the cost effectiveness, you find that HIV vaccine would be cost effective. But also just more broadly, an HIV vaccine would actually relax the requirements of, of going to the facility, which has been a challenge, right? Not everyone wants to be visiting the facility now and then. So it would actually help address that, especially if you find, for example, a vaccine that you can take once a year. So it would limit the number of times one has to visit the facility. But also, most importantly, it would expand the choice of available HIV prevention options. Choice is important. I think we've been talking about, I can't overemphasize enough the importance of choice and why it is important for HIV prevention. So what will it take to find an effective HIV vaccine? I think Barton really laid out like the challenges that we are facing currently, but also the potential solution that could help us take us on the path to see that scientific breakthrough in finding an HIV vaccine. A sustained finding, I'll not go into details on that one. I know there's a discussion around funding and why we need that for HIV vaccine R&D. Renewed commitment, I think, is what we need. And this conversation, I think it's part of renewing that commitment energy from the different stakeholders, so that we can all work together put our efforts together to find that vaccine, but also community engagement. I think we've talked about this long enough. We need community engagement from concept to design to implementation. We need the input of the community to inform, to provide input into what are the needs of the community, because we don't also want to end up developing an HIV vaccine that will just sit in the facility and people are not accessing it. So it's important to involve communities, but also in blessing, I think Pardon mentioned that as well, in blessing the lessons that we've learned, because we've learned so much. Even though we haven't found an HIV vaccine to date, there's so much that we've learned, on that journey, that HIV vaccine discovery journey. There's a lot of lessons that we can draw from the failures, like from the partial successes that we can use that could propel us onto the journey of finding that effective vaccine. And lastly, but not least, improving that strong coordination and partnership among the different stakeholders. Yeah, I think that's what I wanted to share from the community perspective. Thank you.
Maureen, thank you so much. That was a great perspective on the continued relevance of an HIV vaccine and why we need one. So thanks so much. We're going to now turn to our first panel discussion, and that's going to be chaired by Gavin Churchyard at Oram, South Africa. Gavin, I'll turn it over to you.
Thanks very much, Susan, and hi to everyone. So this first panel will talk about what it will take to accelerate early phase clinical research. As we've already heard from these first three presentations, we have a rapidly changing HIV prevention landscape, and this is making us rethink of how vaccine R&D is being done with respect to product development, clinical testing, related efforts, and capacity building. We've heard about a number of new strategies and platforms, and particularly of note is the messenger RNA platform that will allow us to iterate rapidly and accelerate development of HIV vaccines, as well as the perspective of potential end users of a vaccine. And so what will it take to test these strategies to pursue answers and accelerate timelines? A reminder to please post questions to the panel in the chat with respect to what it will take to accelerate these early stage clinical trials. So as soon as we don't have any, I'm going to initiate the questions, but I'll be looking out for your questions in the chat. So Bart, as soon as you started with the presentation, maybe if I could ask you if you could just further elaborate on the benefits, but also what are some of the limitations of using a messenger RNA platform?
Thanks, Gavin. Sure. I think the benefits just emphasize both what Kundai said and what I said, is that we're not going to be able to iterate. We found that out with proteins. Proteins simply take us too long to make to be able to get into the flow of iteration. And so we've done a number of studies comparing proteins in animal models with mRNAs, and we don't think that we're going to be losing anything with mRNAs. In point of fact, the mRNAs may actually, we have data that the mRNAs may actually be better based upon how they're processed, and the proteins that they encode are processed intracellularly and put on the surface of cells that the mRNA LNPs enter. That mRNAs may well be better in selecting for these rare mutations, particularly the mutations that are needed for binding to glycans. And so there may be some actual inherent advantages to the mRNAs in addition to the speed and iteration. What are the problems? The problems are going to be that the LNPs, the cationic ionizable lipid nanoparticles, are part of the magic of the mRNA, and that they are very good adjuvants. They induce high levels of IL-6. They induce high levels of other chemokines. And in higher doses, they can be fairly reactogenic. And so that's one issue, is the side effect. And then the very, very rare anaphylaxis reactions that are thought to be, haven't been proven yet, may be related to polyethylene glycol. So that having been said, the mRNA vaccines for COVID have been given to literally billions of people. And so that's very, very encouraging that the safety profile with the HIV vaccines will be acceptable, but that remains to be seen.
Great, thanks very much, Bart. So Jim, if I can turn to you now, how can we test these products faster, what clinical trial designs can we use to iterate more quickly?
Thanks, Gavin, and thanks to the presenters who provided, I think, a very strong foundation for this discussion. And that's really, the operational question, Gavin, that is before us, and all of us as a community, I think, need to work as best as possible in defining what designs are most strategic and effective in getting the laboratory and analyses out of these clinical trials so that we can make those very strategic decisions to move forward. And as Bart highlighted, optimally, we would have a multivalent vaccine that targets the MPER region, CD4 binding, V2, V3. But we're going to have to work on each of these individually and optimize those antigens individually before we're able to really combine them into such a multivalent BNAB strategy. Therefore, these designs are, I think, optimally focused on individual antigens as quickly as possible. Again, getting the clinical trial launched and implemented so we have the laboratory assays run as quickly as possible, the data analysis completed as quickly as possible, and therefore the critical decision point and in interpreting that data to move the field forward to the next iterative study. And we've looked very closely at our internal operations on how best to optimize each of those steps and have pulled together, I think, as best as possible what we call our portfolio management with regard to these experimental medicine trials. And it really is then foundational for us to move forward with many XMED studies to get the data, interpret, and iterate.
Thanks very much, Jim. So Johan, maybe if you can pick up from what Jim was saying and elaborate further on how the experimental medicine trials can be fitted into the whole product development pathway, and what's the long-term perspective on that?
Thank you very much, Gavin. And I want to highlight the interest of the introduction in terms of understand the picture of, on one hand, the scientific complexity of what we're undertaking, and on the other hand, the end user expectations. And we're also at IAVI very much busy with thinking about the operational pathway to ensure that there are clinical trial avenues that are able to support rapid progress of this iterative endeavor. We believe it's going to be very important to be very transparent with the affected communities and the communities considering hosting trials and the volunteers on the fact that we are not exactly close to having a product that we're contemplating licensure, but that we're contemplating for licensure, but that we're going to go through a pretty lengthy upstream research evaluation. And creating consensus with the regulators for that allows rapid iteration of the clinical trial would be extremely important. And maybe we need to think about creating a clinical trial platform and writing an overarching study protocol that is available to host modular introduction of individual immunogens or testing of immunization schedule changes in the way that is going to accelerate decision-making and implementation of new trials. I think a lesson from COVID has been that with political will and ability to change our ways of working, there is clearly a capacity to accelerate proceedings. And I don't think that we should contemplate having to develop new protocols that have to go through lengthy reviews before being implemented for each time we want to test a new immunogen and a schedule change. So these are, I think, important elements to support rapid iterations.
Great, Johan, thanks for identifying the need to develop a platform type approach with a standardized protocol that we can plug and play with. So then if I can ask you maybe just to pick up on that and share your perspective on some of the regulatory challenges we may encounter with doing these experimental medicine designs. And then I've got another question for you following that around whether comparative adjuvant studies with proteins that, whether comparative adjuvant studies with protein be relevant to the mRNA approaches.
Yeah, thank you for that. I mean, I think the regulatory issues, we're going to need to have some consensus discussions with the regulators, because we have to decide how much preclinical work we need to do before we do first-in-man studies. And so some of the issues around overall toxicity and preclinical work, what kind of immunogenicity studies we are going to have to use, and whether we can have a generic approach to certain platforms that don't require each product to go through preclinical work, but enable us to be able to compare the platform with previous work in that area, so that we don't have to do an immense amount of preclinical work to advance our first in human studies. So I think that's going to be critical. How much, what do we have to do in the animal models before we can get into humans, and some consensus with the regulators to see whether they will waiver some of the preclinical work so that we can move forward. So I think that's going to be very important. The issue around the importance of protein and adjuvants versus mRNA, I think we need to progress both areas, because adjuvants, as you know, are critical for immune responses, particularly if you want to direct an immune response in a certain way. And so I think we're going to have to discuss how we address the issue of adjuvants going forward, and what is required. So I think that kind of approach, Bart, I know that there's lengthy processes to make protein, but we also need to find out, what are the bottlenecks, how do we speed up protein biomanufacturing for GMP material, what are the gaps in the production area as well as in the clinical trial area. Jim spoke about trying to look at the bottlenecks in data management and laboratory, but we can't have clinical trials that take a year to enroll 10 people. We can't work like that anymore. We have to enroll faster. We have to look at all the bottlenecks, and we have to find ways that give the regulators and the ethics committees comfort to advance into humans with minimal preclinical data. We need to look at the bottlenecks in GMP manufacturing, and why does it take us such a long time to get onto the manufacturing lots. We're not the desired customer, and therefore we have to wait months before things happen. So if we are interested in iterating fast, we have to do everything from the cradle to the grave, and that's from the manufacturing to the scale up of enrollment in HIV vaccine trials. And that gets to me the importance of involving sub-Saharan Africa. We're very good. We know the burden of disease. We are daily affected by HIV, and so there's a lot of appetite to speed up the processes in our countries. And so I think we have to look at where we do our clinical trials and how we can work with our regulators to speed up this process. But that comes obviously with political will and the will of everybody to try and make these things work. I also no one really mentioned the mother-to-child arena and the work that we've been doing with Bart Haynes in looking at HIV-exposed infants, and using these approaches to induce body neutralizing antibodies in babies to prevent breast milk transmission, which may be a very important aspect to this advancement. And so I think for me, it's about looking at every area of the from discovery to application to make sure we get rid of all the wastage that we see at the moment. There's a lot of we're lethargic. In our way we conduct our trials. We're expensive and lethargic, and we need to change that.
Thanks, Genda. You need to have this, tell the TV field the same thing. Bart, I'll come back to you with respect to what Genda's raised and some other questions in the chat for you. If we can now move on to Nandi. So if you could please share with us your thoughts on how we could collaborate. What are the key partnerships that we need to establish to accelerate these early phase clinical trials?
Thank you so much, Gavin. I think for me, it's all about bringing in communities along the journey. So for years, I worked for the Orem Institute and then at the HVTN and now at AVAC. And research literacy has always been something that has helped us make communities understand the importance of research and understand how to engage in research. But now we actually have to build implementation research at the same time because we've got new products. And this presents opportunities about how products get approved, how products are implemented and rolled out. So we really need to work with communities to understand that there's a lot of choice and options out there in the field right now. So it's not only about what the trial is about or what kind of study one can join, but it's about the trade-offs between being in a study as well as being given the opportunity to actually access a proven product. And it doesn't mean that these new products don't need to be developed. In fact, I think it actually reinforces the reason or the argument for newer products being developed because current PrEP options and future PrEP options are going to leave a huge gap in the market for other products. So I think for me, as much as we need an HIV prevention vaccine, researchers need to understand that communities are grappling with the idea of here and now. So, and part of here and now is demanding access to products that already exist. And HIV prevention, as an HIV prevention advocate, we are deeply committed to accelerating access to research opportunities, but we need to do this in the context of rollouts, as well as in the context of delivering what we already have, while we develop what we need. And that's not, and that's going to take a different degree of engagement and a different degree of literacy. Oftentimes, when people think about community engagement, it's usually tokenistic and at times not strategic enough. I think that we are learning in the field right now, especially in the process of, with the process of research to roll out, that it's really important and crucial to involve and work with the community, civil societies, and advocates to inform how we move forward. These primary stakeholders have been helping inform implementation studies so that researchers are getting the right information out of them. And that helps with making sure that there aren't any delays, making sure that the research moves from research to rollout, and that product developers are being pushed around issues of pricing, transparency, and working with the medicines pool, patent pool on what generic manufacturing should look like. These are all very crucial milestones that are happening now, which have been built on a backbone of communities, civil societies, and advocates being engaged throughout the R&D process. So it's not just about getting potential participants enrolled in an HIV trial and getting that research done, but it's about the end game. So researchers need to bring communities, civil societies, and advocates along the journey of developing an HIV vaccine, which is a really important journey. Researchers need to understand that communities want to be on the journey of to find an HIV vaccine, but it's only a subset of the journey that communities are really on. Communities are on a much bigger journey. We need to know that there's a prevention roadmap to 2030, and researchers have to remember that communities are hearing that we can end HIV as a global health threat by 2030. That's a UN target, that's what PEPFAR has signed on, and that's what the Global Fund is driving towards. So the journey is bigger than just developing an HIV vaccine. It's about the bigger picture. It's about the bigger picture end game. We need to understand that we are advocating for researchers to collaborate with communities who are in different places in this journey. Again, it's not about recruitment issues. It's about engaging communities in supporting research. The COVID-19 experience taught us that HIV researchers need to conduct HIV research faster. We have also learned from COVID that even researchers, as researchers get to the end of the journey in terms of product development, that having the final product does not end the epidemic, because the issue of delivery, access, and hesitancy. The point that I'm trying to make is that the important,
we need
I'm
to
almost done.
Okay.
Just the point that I'm trying to make is that we need to bring communities along to developing an HIV prevention vaccine, and that researchers need to show that there's an end game, or there are next steps, a series of next steps after the products have been developed, to ensure that there's confidence not only in the trial but in the research enterprise as a whole. I think I'll end there for now, but I would like to emphasize the importance of just research literacy and making sure that you bring communities along the journey of the R&D process. Thank you.
Great, thanks very much, Nandi, for stressing the importance of taking community along. So, Isaac, if I can come back to you with, can you just share your thoughts on what infrastructure requirements will be required to accelerate early phase clinical trials?
So, Gavin, it's for me the question, sorry.
Yes, for you.
Okay, so thank you. So first of all, I would save time because I agree with all my colleagues and many points were raised and very valid points. So I don't have many things to add. Regarding the infrastructure, I think we have to reset what we are doing for the vaccine, because we have new technologies as mRNA very well presented by Bart. And we know also the problem of the manufacturing and GMP to have some protein. And we are convinced that we need to combine both strategies and to have a prime or to have a boost. So the question for us is how to combine these different things regarding the multiplication. I think the complexity of the field regarding the number of antigen design, the number of candidate vaccine. So in my view, we have three big pillars. We have this antigen design and the new delivery of the proteins and the mRNA and the new candidate and the combination. So we expand completely the number of combination and regimen we may test. We have the need to go or no go based on preclinical studies, as Glenda mentioned, to go or no go to some experimental medicine. So we need, as Johan said, we need some protocol or generic protocol or something very easy to move new candidates or new combination to the experimental plan and to discard or to select. And the next step will be the go, no go based on what the laboratory assays. And I think in the last 10 years, based on the data of the LV144, we have made progress in terms of evaluation of the immune response and to try to track exactly what we want as the immune response. The problem for us is the multiplicity of the candidates we have today, the design and the go, no go decision to go step by step, and of course, the resources to have the manufacture to the different products. And thanks to Kande, because Kande showed very nice review of the trial, but we may complete this because we have, for example, in Europe, 2 ongoing phase 1, phase 2 trials with original design, so which add also some complexity and multiply the number of candidate vaccine we have. For example, new delivery of the protein vaccine with the dendritic cell targeting, which is a strategy in phase 2, or DNA replicant, which could be very interesting combined or to see with the RNA or the combination with the protein. So in my view, the infrastructure would be rather instead of infrastructures, because production of mRNA, production of protein, there are specific issues. Most importantly for me is how all together and under the umbrella of the global HIV vaccine, we try to go further to these go, no go steps and experimental design to discard very quickly the iterative process to select and to test different combinations. And this is what we need, because honestly, since we have a candidate in our different consortium or based on the science we have done in the last years, we are going with one clinical trial. But the problem will be to combine or will be to compare. So we need perhaps to put, I would say, brain, resources, and all together to think what we will do with all these candidate vaccines coming in the field.
Great, thank you very much. So if I can come back to Bart now, there was a question around that messenger RNA vaccines, apart from having an adjuvant effect, also have the property of delivering proteins in a more prolonged manner that are probably of interest. Is this property that mimics natural infection more necessary or a determinant in the induction of broadly neutralizing antibodies?
Well, thank you for that question. It's a very important question, and there's several components of issues buried in that. Number one, you mentioned the adjuvant, and that's an important part of it. But the second part is how it's processed. Is it more like a virus? As I mentioned, the mRNAs appear to be better at selecting for the right kind of mutations and giving rise to antibodies that can either bind completely to glycans or bind to glycans plus polypeptide backbone, such as for the V3 glycan type of VNAbs. We are wondering, haven't proven yet, but if the glycosylation is not different, the sugars that get put on the proteins are not different when it's from an mRNA pipeline within the cell versus when we make it in 293 cells and then give it to the individual. So that is being studied. So it may well be that there is some advantage to how it's processed, et cetera. Yeah.
Thanks, Bart. Glenda, if I can come back to you now. So Kundai mentioned that most of the phase 1 studies are being conducted in the US and that we should try and conduct more studies in other continents. And you echoed that call to really utilize the clinical trial infrastructure in Africa, not only that it exists and that it's good, but that we also have the burden of disease and that we are willing and able. So do you want to elaborate further on that, please?
Sure. I think that's the discussion that we need to have with the communities and the regulators, and that the rationale for not doing first in human studies or experimental studies in high burdened countries. So I do think we have done it in PMTCT in our with Bart. We went straight to infants before going into adults. So I think we need to look at our populations. We need to look at our regulators, our communities, and our ethics committees to address what are the concerns about doing first in human trials in certain parts of the world. In South Africa, we do first in human, and we do experimental medicine work. And I think if there's any, and I think that the hesitancy, we need to understand a little bit further. In the past, it might have been imperialism or neocolonialism or fear that there was experimentation on more vulnerable people. But I think that it's important to discuss this with communities, with regulators, ethics committees, and scientists in countries to see what are the barriers for expediting these kind of trials in places where you would not normally do them first. And I'm not talking about South Africa in this case, because we have been able to do this quite successfully.
Great, thank you. So I'm going to now ask, there's a question about local manufacturing capacity, but just manufacturing capacity in total or overall. So Bart, do you want to maybe just kickstart this discussion by talking about some of the manufacturing challenges, and then we can talk about what are the opportunities for local manufacturing, and then please, it's open to the entire panel to comment on any aspects that has already been discussed. Bart?
Sure. So very briefly, several big challenges with manufacturing, not the least of which is raw material, supplies, leading to massive delays. As much as we can bring in-house, we are, because the field is jam-packed with regard to needs for toxicity studies and all the other regulatory kinds of things that routinely get farmed out and take months to years to get scheduled and completed. The point about the platform that Johan made, I think, is really important. And part of the success in getting the bivalent COVID boosters out is that the FDA had already seen everything in the BioNTech, Pfizer, and Moderna platforms, and the only thing that changed was the insert. And so getting to some kind of homogeneity in the platform so that it is a plug-and-play manufacturing kind of thing, getting through to release and through the IND process is important. With regard to local manufacturing, I think that that's very important. And we are at Duke, we are committed to global access and have shared all of our mRNA know-how with Afrigen, for example, in South Africa, and with others in the United States to create not-for-profit hubs, if you will, to be able to get this work done and to collaborate. We're also talking with folks in Thailand with regard to sharing our information there. So I think it's really important to share. It's really important to get local production up and going as robust as possible, to deal now, while we're working on the enabling technology, to deal now with the delivery issues once they arise.
Great, thanks, Bart. Beatrice, you want to make a comment?
Yes, a very quick one. Thank you so much. Can you hear me now?
Yeah, we can hear you.
Yes, I just wanted to ask you, please, to put the Latin America back in the map here, because we couldn't see us yet. And I just wanted to reiterate that we have a very good local capacity to do studies in several of our countries. And also we have high incidence in very vulnerable populations of young MSM and trans women. So just wanted to put our desire here to be able to be included in future studies. Thank you.
Thanks so much, Beatrice. All right, it's now open to the panel. If you want to make comments on what is already, if you want to add to anything that's already been said, please do so.
I think the production issue that Bart raised is the elephant in the room. And I think that we probably need find a way. And I do like the fact that the only way you could get around it, Bart, was to in-house it. And I think that that's an excellent strategy, because we don't have to wait in queues for tox studies and production lines. So I think we've underestimated that part of our pipeline and how long it does take to get onto production lines, and in-housing production or having a process that expedites this, I think, is going to be critical for this iteration that we talk about.
Great, thanks, Glenda. Any other comments?
Yes, Gavin, if I may, regarding this issue of repeating toxicology study, mostly when we go with a new vaccine, even if it's the same platform. I think the discussion we need to have, exactly as Bart said, for the COVID-19, since the same platform, the only insert was changed, so there is an acceleration. I'm not sure regarding HIV, when we change the antigen design, that we will benefit, I would say, from the same exactly process and the acceleration based also if we have the same platform. In our experience, for example, even with the same vehicle in the same platform, the change of the antigen, the design could be also considered as a first in human, even if you use the same platform. So I'm not sure that today the regulatory agencies are really prone to move exactly what we have learned from the COVID-19 to other antigen design, even if we are using the same platforms.
Great, thank you.
I'd like to add a comment on the, we've heard the importance of local involvement and local capacity. And I think it has to be considered really on an end-to-end perspective. It's not just about manufacturing, and it's not just about access to product. It's really creating an end-to-end environment that is supportive of vaccine development capacity. IAVI has been engaged for years in building relationships with investigator networks in Africa, and indeed totally supports the notion that we have to move fast towards the affected community and the affected areas. Mm-hmm. As we approach proof of concept, we have to really apply a downstream perspective about what it will take to have these access considerations sorted out very early, and it's very good that there is an expanding platform of mRNA creation technology. For the moment, there is a very low number of players that are able to provide access to their technology for research, but it would be much better to have a broader panel of accessible platforms in the future.
Great, thanks. John, do you want to address the final question, which is, how do we address the IP issues that delay tech transfer? Any thoughts on that?
Well, thanks, Gavin. And I was just thinking, as Johan was speaking about this, the critical need to have industry engaged in this activity. We as a community need to demonstrate the potential of the induction of BNABs and demonstrate the concrete evidence in which that can be accomplished. And I know Bart at DHVI and even Moderna and BioNTech have talked about how they can individually, as production entities, address this elephant in the room that Glenda mentions, and how we engage with them is going to be critical, so that if there are IP issues that are critical to be maintained, there needs incentive for these partners to be engaged with us in anticipating the scale up of material, even for the phase 2B and phase 3 type of studies that are hopefully coming in the not too distant future. So that is a very complicated negotiation with regard to tech transfer and agreements. If we're serious about transferring some of this to the countries most impacted, and I know many of us here are committed to ensuring that happens.
Right, thanks. And thanks to the panel and to those of you who posed questions from the audience. This was a very rich discussion and will lead into the next session around unpacking some of this in more detail. So a big thank you to everyone. So we're now going to move into the breakout rooms. Elena, who's going to do that?
Thank you, Gavin.
Right.
So as you said, there was some very important point made by the panel during the discussion. What is going to happen now? We are going to allocate the participant into 3 different rooms, and in each room we'll discuss a particular aspect related to the product, their prioritization, development, selection, or we will talk about how to accelerate the clinical aspect of the research. And in the last room, the discussion will be around capacity building. In each room, you will have a facilitator who will lead the discussion. You will see already a list of bullet points to support engagement and to trigger the discussion themselves. There will be a rapporteur who, after the break, will summarize what has been said, and you will have an IS staff or myself to deal with room issues. So it is 11 past whatever time it is where you are. Thank you very much, everybody, for rejoining part 2 of the Vaccine Enterprise stakeholders meeting. The agenda is on the screen, thanks to Elena. We will, in the next 1 hour and 45 minutes or so, hear from the rapporteur from each session. They'll have 3-4 minutes each. We'll follow by a general discussion on the point raised in each of the room. We will then move to the final part of this event, which is the panel discussion that brought a number of people, where we talk about how can we make sure that we actually deliver on accelerating research. So I'm going to share my screen, take some notes again in the background to support general discussion that will follow. And I'm going to ask for rapporteur from room 1 and Suzanne to tell us about what was said.
Yeah, thank you, Roger. Hi, everyone. I'm reporting for the first panel. And I want to say, first of all, that we had a very lively discussion. And I think we all agree that good science is going to be critical to motivate industry to engage and for us to achieve the best HIV vaccine approaches. There was also a lot of discussion in the room and concern that that we diversify our approaches, and it's pretty much was broadly agreed that it's important to the field to, not only focus on BNABs and tier 2, achieving tier 2 neutralization, but to really learn from decades of HIV R&D, as that was pointed out by one of our speakers, with other vaccines, and take a comprehensive approach that includes both B, T, cell responses, as well as innate immunity, and to exploit the biomarkers that have been defined by previous proof of concept trials that have been done in humans. And I'll mention those. We're looking very, very intensively right now at the EMP results, but we probably shouldn't ignore the other results from the other trials, such as RB144, HVTN 702, 705, 505, and STEP. I mean, there are results there that we should pay attention to, and that was kind of the overriding consideration. It was also brought up that we should take perhaps a more industrial approach to take concepts to the clinic fast, to have very strict no-go criteria for advancement from early clinical to clinical and throughout clinical development, and that we should be in a mindset where we kill bad vaccine concepts that have low probability of success early rather than later. In addition, a last point I want to make is that it was strongly believed that we should take the totality of the data, both the preclinical and the clinical, and optimize the clinical models we have at our disposal to down select immunogens and approaches, be very critical of ourselves along the way. I think that's it for our session. Thank you.
Thank you very much, Suzanne. I am going now to ask Sandy.
Hi, everyone. I'm the rapporteur for our clinical trials breakout room, and that was also a lot of lively discussion. I'll divide the 3 minutes, I think, into 3 categories, which are trial designs, assays, and then a little bit on manufacturing. I think we spent the bulk of our time talking about trial designs and implementation. When we talked about the fact that what had come up in the main discussion before is a question about first in human trials, and talked about the fact that the bar is moving and that there's progress in welcoming these trials in LMIC as a right, whereas before there was more concern about moving more slowly and conducting early safety studies in the US or Europe or what have you. But to be able to do this and push this out to LMIC, we need to rely more on standardized adaptive trials wherever possible, so we can minimize the time that's required for each new protocol development, so we can plug in experimental products into the same platform, and the HVTN is just completing that, their standardized protocol platform for that. This also requires some common safety standards that was raised by another speaker in the room, as well as educating regulators in different countries about these concepts so that they're better received when we bring these protocols out. Because it was raised that there are trade-offs between time to regulatory approval, which often takes much, much longer in countries outside the US and Europe, and time to recruitment, which often can go much faster than in some of the US sites. And so then turning to issues with recruitment, it was talked about that it's very helpful to develop volunteer registries. Susan raised that the HV10 is moved to not limiting participants in phase 1 trials to those who are low risk, but then it was raised that, of course, the trade-off there, the caveat is, of course, thinking about VISP and risk to people who may for which that might be a bigger issue to continue to test positive on an HIV test, which then is a good segue to the assays. Of course, we talked about investment in developing, continued investment in developing capacity for assays, and that the commonality of assay readouts will allow us to be able to compare across different trials. But the trade-off there, of course, is some of the most specialized experimental medicine trials may require some very specific and highly specialized assays, BCR sequencing or what have you, that, can't be widely implemented. And so there would have to be a prioritization of capacity development. And in that context, also, it was raised, the counterpoint that there still may be a value for conducting some of the earliest phase trials locally in 1 or 2 countries for those reasons. And then to support all of this, of course, that was raised that there can be requirements for shipping and things for some of these specialized assays if you're conducting them in real time. So these are the trade-offs we have to think about depending on the complexity of the trial and the tiering of the assays. And then lastly, there's a talk about manufacturing, which may go a bit into the next breakout room, but you know, that as was discussed in the main session, there are similar backbones may help speed approval when the inserts are changed of well-tested platforms without the need for tox studies. But of course, if we're switching inserts between pathogens, that remains to be seen, where a well-tested platform in the COVID space, for example, whether or not that can just move to HIV without requiring additional studies. And then lastly, there is, of course, a lauding of the expansion of local you know, of mRNA manufacturing capabilities. There's also a caveat that mRNA manufacturer or mRNA vaccines as a whole don't always, you know, necessarily guarantee the quality of the protein production post-transfection. So that's a caveat to be watched. And I think that concludes my 3 minutes.
Thank you, Sandy. I'm going to hand over to third reporter with William Kilimbi from Zambia.
Yeah, thank you. Can you hear me?
Yes.
Are you able to hear me?
Yeah, yes, we can.
Okay, thank you. So I was in group 3 and the facilitator was Linda Gil-Becker. We're looking at infrastructure and mechanisms to accelerate iterative clinical research. We had a lively discussion there too. I think we sort of broke this down into 4 areas where we discussed extensively 2 of them. That was really needed to look at the lessons learned with COVID-19, aspects of the 3 C's, that is communication, coordination, and collaboration. We also wanted to look at the critical importance of data and framework, data sharing and frameworks for regulatory approvals, as well as placing all this in the context of in the context of ARV interventions. So in terms of lessons on COVID-19, really the extent of funding for COVID-19 was, so a lot of money had been thrown to COVID-19, yet we've seen that funding for HIV has sort of plateaued. And therefore, development or research and development in terms of HIV vaccines is perceived as quite a risky area. We need to begin to think of de-risking this industry. We think well thought out pipeline for development, manufacturing for the clinical product of the IP, and also scaling up manufacture once we get the efficacious vaccine in place. All these need to be thought out critically. It is also important that data information is shared to accelerate decision-making on efficacy, and we need to develop platforms for this in readiness for that. With COVID-19, there was not a lot of thought given to access of vaccines once the vaccine was developed. And this should be a priority as we think of developing an efficacious HIV vaccine. Also from COVID-19, we also learned that the platform approach allowed comparison of data and results, and this enhanced decision-making in terms of emergency approval for the vaccines to be scaled up. Collaboration by involvement of different regions of the world is also another aspect that came up in the previous session. And we need to think about all the different areas, and we should not forget any areas as we develop a vaccine. In the context of community engagement and involvement of the community, it was thought that this is a very critical aspect, and we need to think about how we involve communities better, and how we improve our capacities in doing this to ensure that once the products are tested, there's a lot of community engagement right from the design of clinical trials and all other aspects related to good participatory practices. We've conducted several efficacy studies in these. By and large, these have been testing one product at a time, recently with large efficacy studies. But we need to think about community engagement, crucially in the context of conducting experimental medicine trials. The other aspect that we see from COVID-19 is simultaneous testing of candidate vaccines. We need to rethink these new technologies that are being discussed now, understand the basics in terms of immunology. Again, this is a risky endeavor, and we shouldn't focus on just one modality. Rather, we should give all modalities equal attention. And we also need to appreciate how much the COVID-19 pandemic and vaccine development research and development benefited from HIV, and learn from some of those lessons, emphasizing the point that the work that has been done in HIV is very crucial. For instance, you may be aware that HIV vaccine trial helped develop some of the COVID-19 vaccine trials that have used the adenovirus vectors. And this obviously de-risked COVID-19 vaccine development. Also, the potential of using common DSMPs, common endpoints, correlates of protection and inclusion-exclusion criteria from the outset, and also inclusion of different key populations facilitated vaccine development, as well as enabling us to compare data sets from different areas. As related to the 3 C's in terms of collaboration and communication, it is key that shared communication to address aspects for go or no-go decision-making are thought very carefully about, as well as shared understanding of targeted profile vaccine. In this regard, we need to think of really what do we intend the vaccine, or what is the characteristics of the desired vaccine to allow everyone to work towards that goal. Need for more basic immunology and to understand HIV immune responses a lot better. Very common to have strategic development plan in the manufacturing area, but this has not been done very well for HIV. So there should be consensus building by developing strategic plan, roadmap, as well as think of this in the context of the new vaccine design platform. And this sort of strategic planning is suggested to be housed at the Global Vaccine Enterprise. Communication to include industry. Industry is not going to initiate this communication. And again, it's being suggested that perhaps the Global Vaccine Enterprise should initiate this communication with industry so that we can anticipate barriers and be able better prepared in terms of addressing those barriers. We also discussed very briefly on other capacity building, enhancing lab science, particularly in capacity building of local scientists where burden is highest, and also to think about very carefully how we sustain this capacity that is built in particularly low middle income countries as they continue to participate in HIV vaccine research and design. Thank you. I'll end there.
Thank you very much. You have run out of posted space just in time. Let me go back to that. So we now have about 30 minutes to continue the conversation as a group. We have about 50 people on the call. The way I would like to propose to do that, you have now on the screen various suggestions and points raised in each of the room. And what I would like to achieve with this conversation is a possible crystallization around a number of priority things that needs to be done, things that needs to be done differently, things that needs to be looked at more closely, but also going maybe one step further in how do we actually move to the next step for many of the points that have been made. And if I take a few more learning from past study, how do we make that happen? Simplifying regulatory path for product, how do we make that happen? Can we agree? Is it possible that people in the field will agree on a standardized trial and build common platform for clinical testing? How do we develop new ways of collaboration and increase capacity, especially in LMIC? And these are the kind of idea. And I'm just going to open the floor and really give you the floor to make your comment, and I'll jump in when I think we need to move. So also what we can do, if anybody has any comment to add on that, disagree or agree or want to support or to add, please do come on screen, put your cam so it's easier for me to see who wants to speak and give you the floor. Everybody, yeah. So who would like to start? I will pick people in my room for a start. Oh, it's a big silence. Ah, there is a hand raised. I can't see who raised their hand. Let me go back up. Yes, Roger, go ahead.
Yeah, so I apologize because I have a limited capacity to have innovative ideas to comment, but I may try to start by rephrasing a bit what we discussed during our room session in different themes and some addings to that. So I think one of the first key issues, at least for European researchers, is the capacity to have a sustainable funding for this research. So we have 5 years program, and then when we end this program, we need to start from scratch again, and we start doing additional consortia and working with additional deal because the cause for the development has changed, the phrase of the cause has changed, so we need to adapt itself. And for this, I think what we need is a strong advocacy to the politics in order to encourage them to make these programs more sustainable, because 5 years program is not sufficient enough to go up to the experimental medicine trials. The second point is that what we already discussed in earlier sessions with you, Roger, is that how much we can learn from COVID-19 and the reverse. One of the key points I see beyond the fact that there was big money on the table very quickly for the development is that most of the successful products have been de-risked before by academic research or small companies, and in many cases thanks to HIV research. So HIV research has provided innovation because the scientific challenges that is required for the development of these very complex products, and then brings new ideas, new platforms, new antigen design process that was used for rapid development for the COVID-19. And this academic research has brought these vaccines, the HIV vaccines, up to what we call pre-industrial level, meaning that many things have been de-risked in this case because there were preclinical work done, there were toxicology work done, there were clinical trials done with this platform, and we were already improved the process for bioproduction. So the companies has just taken these available strategies and had to adapt to the COVID-19. So that's for one. And saying that, that has two consequences for me. The first one is that we need to improve probably the capacity to have a very agile and very organized R&D pipeline from antigen design to production of the vaccines, accessible to academics in a fast way. So meaning that it's not only a matter of technology and a matter of science, it's also a matter of management and organization, meaning that in this case, we need to have already prepared contracts, agreements, and whatever to go from very quickly from the preclinical to the clinical and to the bioproduction systems in order to go quick to the clinical trials. The other consequence is that we may try to engage much earlier private companies in the vaccine research if we convince them that the investment they may do for these very challenging vaccines actually will benefit the development of other type of vaccines. And then they can try to have a return from their investment by developing the vaccine for the platforms for other purposes than the one they invested in first for the vaccine research. And I think for my comments, I will stop here.
Thank you very much. So I'll take the point on, one of the points you make is better organization and a more streamlined process. And we find that in a number of posted here when we talk about standardized trial, about working more like the industry, down selection of product. And what's interesting in the room I was, the product room, there was a call for having more approaches. And at the same time, there was a realization that it was then how do you go about doing the down selection and agreeing go, no go criteria, and that it was thought being something very difficult. Does someone want to comment on that? Or Suzanne, if you want to add to
what was here, I was actually just writing a couple notes on go no go criteria. So one of the issues is that for instance, the HVTM program, where we have a lot of these B-cell immunogen studies going on in our XMed program, they actually have very different objectives within them. The primary objectives of these B-cell immunogen studies are not all the same. And, one thing that we were asked is, what would the criteria for success be, which, can translate into go, no go criteria as well. But I don't think you can have a sort of a standardized set of go, no go criteria, because some of the studies, the primary objectives are to induce a specific germline. Others are to drive a specific lineage, and others are yet to actually induce neutralizing responses. So, it is complicated in that you probably have to keep the go, no go criteria tailored to the specific product or regimen. And some of them are even just designed, the go, no go is not to move it from phase 1 to phase 2 necessarily, but to say, yes, this product induces the proper germline or lineage, and now we can include that in a regimen where we're adding an additional product to shepherd us towards the neutralizing response. So I was making notes on that because it is a complicated question, and there's not a one size fits all.
Yeah.
So can I comment on that? So John, from a each trial perspective, that's true, but from a portfolio perspective, when you decide what to put in your portfolio, which lineages that fulfill their criteria in their early phase trials, when you start combining things, is there an effort now to work backwards as to what's the high level profile of the combination of immunogens that you'd want to put in an eventual vaccine, and can this be driven by knowledge of the breadth and potency of the antibody, the BNAbs for which you're trying to induce by active vaccination? So are you having those discussions? Maybe you can answer that, or somebody else in addition at the HVTN. How are you seeing that, in terms of portfolio management perspective?
That's a good question, Susan. I think I've been in side conversations about that, but maybe if Jim is still on, he can weigh in more on, if there've been, more formal discussions on that. Not so much as far as my understanding.
Yeah, because I know we're at the early science stage, right? And like for VRC01, we may want to make that, but to just learn. But what do we really want to make based on the combinations of BNAbs you're looking at for passive administration? And how will those two things fit into the whole big picture? Thanks, Jim.
Then Eve.
Yes, thank you. If I may, just to follow that, I fully agree that there is some candidate vaccine or strategy hypothesized driven. So it's obvious that the objective of the trial is to demonstrate the proof of concept, as you said, for example, the germline targeting or the immunofocusing or the neutralizing antibodies. This is very important because, of course, we design the candidate vaccine and we have the objective to demonstrate the hypothesis. On the other hand, if you have a vaccine that today induces polyfunctional CD4 T cells or the high level of binding antibodies or anti-V1, V2 or IgG3 or perhaps some CD8 T cells, which are also what we learned from a huge work for the 15 last years in terms of possible correlate of protection or at least associated with some protection. You cannot discard this strategy because you don't have exactly what you expect with some specific design of specific vaccines, which were designed to demonstrate this. So I think we need, as Suzanne said, as a portfolio, we cannot discard a vaccine which doesn't induce directly neutralizing antibodies because they are not designed perhaps to do that, because we don't know if the correlate or the markers associated with the only protection could be perhaps interesting and could be associated with other candidate vaccines. So we need both, as usual. You know, after the failure of the STEP trial and the other, we said that we should come back to hypothesis-driven and proof of concept, but also to try to reproduce also, perhaps also some very interesting or promising results we had in the past. So we should also capitalize on what we have learned from large trials and just to try also to see if we can reproduce that.
Thank you,
in my view.
Jim, did you want to add something?
Well, I think, you know, what Susan was getting at in John's comments earlier are the reality of these parallel programs that are focused on the induction of BNAbs, but through an approach that that is quite distinct from one another. And so we are getting to a point where, not yet, but I'm expecting some agreement on what some of these critical go, no-go criteria will be for reagents to move into more product development. And, you know, I think that there's synergy to be gained from both the passive and active immunization strategies that have been deployed and that hopefully will be deployed in the near future, that will strengthen that synergy, that will help inform what these levels of BNAb induction via these various pathways may be required for actual prevention of HIV acquisition. And that obviously requires additional work in the passive immunization, monoclonal antibody infusion approaches to help inform the active immunization BNAb induction program. And I think these are critical activities and synergies that we'll see mature in the coming years, such that 5 years from now, we will know what the critical go, no-go criteria will be for a reagent to proceed into product development.
Thanks.
I hope that, yeah, does that answer your comments, Susan, and question?
Yeah, Jim, thanks. Yeah, exactly. Thank you for that perspective.
Thank you. So moving on, being conscious of time. So a lot of discussion on product and on platforms and really finding ways to move faster. And at the same time, there was a need for more approaches, but hitting the one that have low chance of probability. Interestingly, and I want to come back to the lessons from COVID-19, we had a workshop 2 days ago on biomarker of efficacy for HIV trial that could be used in the context of PrEP, which is making trials more complicated. And one of the comments was made, and this is a very naive comment, which I'm making because I know we have a different audience today. It's much more diverse. But for COVID-19, it didn't seem to matter what was the platform at 26, ChAdOx, mRNA. It all led to the conclusion that you needed broadly neutralizing antibody for the vaccine to show some efficacy. I don't know if someone wants to comment on that, knowing the answer, but to explain for people who are not familiar why there is such difference. If someone wants to pick that one. Suzanne, I see you light up.
No, I think you need, so back to COVID-19, you do need broadly neutralizing antibodies, but even when we're getting them, we're not protecting against infection, we're protecting against disease. So I think, and I believe in, there's probably experts on the call, that there are other components that are not as well understood, such as T-cell immunity, that are contributing to the protective effect against disease.
Great, thanks. And I don't know if Tom Hanks.
I'll add on to that yeah, even in the COVID space, there's now a look at mucosal administration to induce IgA to try and get closer to sterilizing immunity. And so in some ways the paradigms apply between differences between prevention and control, and so I think even though we were lucky that the earliest correlate for protection against disease was neutralizing antibodies, there's still that same interplay of the different arms of the immune system. That's always going to be more complex with an integrating retrovirus.
Also, I see your cam lights up. Did you want to comment? You are on mute. Okay, so I guess that was talking about developing capacity. Few posted here on developing capacity for essays, but also a lot of suggestion on increasing literacy and knowledge of research, whether it's from regulator or the communities, or even scientists who are not necessarily working in there. What people think is the way forward to make that happen. We know that there are already initiatives, like, for example, the EDCTP in Europe, but how are people doing it on the ground, and how do they propose making it, I wouldn't say improving it or making it better, but to do more of that? If someone wants to comment on this particular aspect.
Roger, LGB here. One point that was made beautifully by Margaret right towards the end and endorsed by Pervin, but I also heard Bart saying it earlier, and I think it's an important one, that I think we can overcome some of this mythological thinking that early phase trials can't happen in, low and middle income countries, or where, typically this kind of basic science isn't necessarily happening, but where the burden is highest, because, it's not acceptable, it's not, regulators don't allow it, don't understand it. One way to overcome this might be to take that science there, right? And just to make the beautiful case that Bart made, that with the transfer of the mRNA technology to Afrigen in South Africa, we will actually be taking that, the product that Petra and her colleagues have produced into humans as a first in human sort of concept in South Africa. So, I think it's a really illustrative point that if some of that transfer happens, and it's important that we do it now and do it urgently, that will facilitate not only, increase manufacturing capability, increase the sort of global footprint of people trying to get this right, but we will also improve clinical trial acceptability around these new products in many parts of the world. So just to make that plug, I think it was well stressed during our phase.
Thank you, Linda. Does anyone want to comment more on capacity building, where to start, what to do, and how to do it? Roger.
One more quick comment on that. Oh,
John.
I think somebody else had their hand up. And mostly when we talk about capacity, we're talking about capacity building in other countries. But I think it occurred to me, we should also acknowledge that to conduct rapid trials where the assays are being conducted in real time, to allow the iterative process to occur, even for the laboratories in the US that are well set up, they don't necessarily have the capacity to do that and may require increased capacity, because once you have 12 to 15 concurrently running studies at the same time, which is what we're developing and we're heading towards, in order for a lab to be running samples in real time, they need more capacity. It is simply not as efficient as waiting till you have all the samples from a particular time point and you can batch them, or batching longitudinal samples of multiple participants. It is less efficient and therefore requires higher capacity in the labs.
Thank you. Next three speaker, Roger, then Stacy, then Katrina.
Yeah, very quick comment. So just to emphasize what's said by Bartolomé in his talk, actually. So having the capacity to do clinical trials is not only having the capacity for the vaccine itself, but it's also for the bioproduction. And this is one of the big bottlenecks we have in many cases for going further into clinical trials. So we need to improve the capacity to produce different type of experimental vaccines, and that it's a very important challenge, I think.
Thank you, Roger. Stacy?
Hi, yeah, can you hear me okay? Okay. I think just building on Linda Gale's comment, and I was in the group and we didn't get to this point, but I did want to just highlight the Matrix project, which is the USAID funded project that started last year. So it's about to start its second of 5 years, which is testing, first in human kind of next generation PrEP and kind of MPT products. And the whole sort of concept with Matrix is to do this sort of in parallel in sub-Saharan Africa, as well as in kind of the global north, and specifically looking at South Africa, Zimbabwe, and Kenya. So I just wanted to point that out, that there is actually, I mean, Glenda made the point earlier that South Africa is doing this, but I feel like there is really sort of a growing body of first in human clinical trials that are happening in, places outside the United States or the global north, and specifically in sub-Saharan Africa. And not entirely sure what the parallels might be at kind of a practical level with the vaccine space. But certainly with the kind of stakeholder engagement piece, I think there are a lot of lessons to be shared. For the Matrix project, we just conducted 3 national level consultations in each of the countries that I mentioned, and got some really, I think, really interesting feedback from across the board stakeholders on the Matrix portfolio, and even just sort of how to engage these countries at this stage. And just to say, I mean, Matrix, I think the first clinical trials will start next year, but most of the portfolio is still kind of preclinical, or even considered parking lot. So they're really thinking about how do we do this sort of product prioritization, and they're really kind of at that stage, which it feels very parallel here.
Thank you, Katrina.
Thanks. I just want to make a point about building capacity in low middle income countries. So one of the things that we've done quite a lot is public and participant involvement and engagement activities. And we've done that in the UK and also in Uganda, and found it to be really very successful, particularly if we do it at the design stage, and specifically asking people who might wish to be involved in research, or those people who are part of the community that may benefit or be affected by research, what they want to see from the research and how we can design it better to meet their needs. And that has really positive feedback for recruiting, which is very important to do these studies at pace, and also for sustained programs in different areas. So that's some experience I wanted to share.
Thank you. There's a few more minutes, and there's two points I'd like to raise. And the first one is on development plan. That's one of the green stickers. There is a group 3, you discussed the need for strategic development plans, roadmap. We heard earlier that it seems it's going to be difficult to get a an agreement on go-no-go criteria for product. Do the audience members think it would be possible to one day reach a high-level strategic plan? And I'm thinking, that in 2003, this is what the Global HIV Vaccine Enterprise was set up for, to coordinate and have a plan. We are 20 years later. I wasn't there then, but we have some people here on the call who were there. What's the feeling 20 years later? Is it possible? Do we need strategic plan, global strategic plan and roadmap? Difficult question. Someone want to pick that one? I see Bill is.
Right here. I would say that if we don't make such a plan, you think about what the counterfactual is. What happens if we don't make such a plan? I think we owe it to ourselves to have a high-level strategy. That does not mean we box things or we discount certain things, but we do need a roadmap of how we are going to implement. And resources are not infinite. So we need to have some way in which we can carry out the studies we need to get to that vaccine, efficacious vaccine that we all want, sooner rather than later. We have a lot of different aspects of prevention that are already existing and will soon come on the line. And so doing clinical studies will get more complicated. So we really do need to start thinking about it at a higher strategic level.
Thank you, and I'm glad that you're in agreement. And a couple of months ago, we had a meeting of European investigators that included funder, and some of the people who attended this meeting are also on this call. And I think we clearly heard the message from European funder that if more funding was needed for HIV to do research, this had to be research which is along a roadmap, and that it was the duty of the researcher to develop that roadmap. And a few people were on that call, if you want to comment. And I see that Margaret also agreed for the need to a roadmap. Someone was going to comment. Bill, yes.
Yeah, hi, everyone. Speaking from experience, making a strategic plan that's agreeable to the whole field is a very time-consuming endeavor, and there's a lot of work involved, and often the field moves faster than the committees that have to create such a plan. I do think that at the moment we are at a juncture where it might make sense to have some broader discussions about the big picture, but I'm not sure exactly how to do it effectively and efficiently.
Thank you. Johan?
Yes, thank you. I want to echo what has already been said, to expand on the fact that not only is it necessary and important to do, but I also think that it's possible. And maybe the expression "go no go" criteria is a bit of a detractor sometimes. Does not completely capture the fact that some of the decision driving points are somewhat qualitative, but I think expressing what are the turning points and what are the the key assays and the key milestones bring visibility, and as such help creating consensus between the many stakeholders that need to engage in such an endeavor and collaboration. It supports sustainability of funding, it supports discussion with regulators, it supports engagement of industry. It goes a long way, and it is absolutely necessary and also possible. And it shouldn't be seen as something that carries all the responses. It's very much as well as about indicating avenues for product combination, for how to host an increasing complexity in a first stage, and then possibly in a second stage, how to simplify
towards a product development pathway. So yeah, I'm very much an advocate of this enterprise.
Thank you. My last point before we move to the next part of the event is really that it all relies on product. It all relies on being able to manufacture this product, on being able to delocalize manufacturing in low- and middle-income country. We know, going back to lesson learned from COVID, that intellectual property has been a big barrier to transfer of technology, to local manufacture, and that there is over 300 patents around each and every COVID vaccine. Is there a role for, well, there is a role, but what role is there for the scientific community, for the community stakeholders, for everybody involved in a search for HIV vaccine to ensure that IP doesn't get in the way of research and on moving research faster? I don't know if we have someone from industry on the call. I thought maybe Carrie was still. Yeah, Carrie is there. I'll put you under the spotlight, Carrie. Talking about industry, IP, and how do we make it possible?
Yeah, I mean, I think that's always the tricky part in terms of, putting together the different agreements and things like that, and it takes a lot of time. I mean, I think, there are ways to kind of around it sometimes. I think, for HIV treatment, some of the like voluntary licenses and other things are some strategies to kind of move things forward, but they take a lot of time and lawyers get in the way. So that's always the biggest challenge is when the lawyers get involved. But yeah, I don't have any enlightening kind of ideas to.
Thank you. I see Tom. Tom.
Hi. No, just, big industries have their own agendas, which are sometimes not necessarily, or maybe often at this point, necessarily compatible with HIV vaccine development. And from our own experience, basically we were slowed down and blocked by a big pharma not being interested and supportive in HIV research. So from this point of view, it was a rather negative experience.
Okay, so I'm just going to close this discussion for now. I'm going to hand over for Suzanne for the next part, to Suzanne and Elena for the next part of the event. Thank you again very much to all who contributed in the breakout room and to the follow-up discussion. This will be very interesting to summarize in a report. Over to you, Suzanne.
Thanks so much, and thanks to everyone for your participation in the breakout rooms and in this general conversation. We're going to move to our second panel discussion, and for that we have Stacy Hanna from AVAC, who's going to lead that panel discussion. She'll introduce the panelists, and then we'll get started. So, Stacy, over to you.
Great, thanks, Susan. Yeah, I'll just jump right into it. I think that this panel session will follow really nicely on the conversations we've just had. So I think everyone can see from the agenda what we're going to what we've got here, and who we're going to hear from are representatives of kind of funding and oversight institutions for this field. So we're going to do sort of a round robin just to kind of hear from each of them first, and then we will open it up to Q&A, I think, really to build on this rich discussion so far. So we're going to first hear from each of these 5 members, just to give us sort of a 2 to 3 minute big picture overview of sort of the future direction of the institution that you represent. And I'm going to go down the list in alphabetical order. And so we'll start with Carl Diefenbach, I think who everyone knows, who is the Director of AIDS at the U.S. National Institute of Allergy and Infectious Diseases. So Carl, over to you.
Thanks, Stacy. But actually, Pervin is ahead of me in terms of alphabetical
spelling
by
or
first
last
name,
name.
by first name, by first name.
Okay, all right,
Sorry.
I'll buy that. Anyway, not to pick on you. So it's my pleasure to participate in this panel. From the NIH perspective, we remain completely committed to the development of a safe, effective, and durable HIV vaccine. From the perspective of the NIH, there are two major remaining challenges in the HIV field. One is the vaccine and the other is HIV cure. And there are probably some intersecting activities between the two, and we have initiated and established programs to investigate and interrogate that space. One of the key factors that may connect the two are the broad neutralizing monoclonal antibodies. We've seen the paper from Michelle Nussensweig's lab recently about how the vaccinal effect was working in people. So I think there's something here that we need to exploit. There's also new findings out of the Scripps Chavid on the way you deliver vaccine. So it's, from my perspective, we're in a really interesting place. We've had in Bakoto end, we are down to just Mosaico, but I think it's really time for the field to step back and assess and reassess where we are, look and plan, but move forward. And the NIH is here to support high priority investigator initiated and other forms of studies that we solicit for HIV vaccines and cure. Thank you.
Thanks a lot for that, Carl. So moving on, the next one will be Carrie Huang, who's the Senior Vice President of Clinical Research for VR Biotechnology and also the co-chair of the Enterprise Industry Partnership Group. So Carrie, over to you.
Great, thank you, Stacy. Yes, so I'm at VR Biotechnology, which is a small biotech company, and we do have actually, the company started about 6 years ago, and I think the Gates was one of the investors in the company early on. And the mission of our company is a world without infectious diseases. So our company is focused mostly on immunology type platforms to address infectious diseases worldwide. So we have a pretty broad program, focused on COVID. So we did develop sotrovimab, which we partnered with GSK. And then we also have a flu antibody program. We also have a hepatitis B and Delta program. And then also the CMV HIV program that was touched on earlier this morning in the meeting. So we are, as a company, committed to addressing a lot of these infectious diseases. From our standpoint, we are a small company. We have good resources, but not unlimited resources compared to some other larger pharma companies. And so we have been working specifically on the kind of the HIV vaccine side, with Gates and with HVTN and other funders to facilitate the development of that program. So I think a lot of you saw some of the initial data from the VIR-1111 data set last week at CVD, and then we will be initiating another study with HVTN next year with the subsequent vector for that. And so I think for us, the partnerships are critical in terms of being able to move these programs forward and being able to leverage the expertise of the different partners that we have. We have some internal expertise on the science and the biology. We work also with Lewis's group. But then we can also leverage, HVTN and Gates and their experience in terms of conducting these large vaccine trials as well. So hopefully, being able to work all together. My other role is as co-chair for the IAS Industry Vaccines Group. My fellow co-chair is Linda Gale Becker, and so this group was kicked off earlier this year. And I think we just had a session a couple of days ago focused on biomarkers for vaccines. And I think what we want to try to do over the next couple of years is really try to engage different parts of industry in terms of diagnostics and other manufacturing, as well as traditional kind of pharma companies to try to facilitate this work going forward. So I will stop there.
Great, thanks for that overview, Carrie. So next up is Margaret McCluskey, who is the Senior Technical Advisor in the vaccine branch in the research division at USAID, I think well known to many of us. So Margaret, looking forward to hearing from you.
Thank you so much, Stacy, and greetings, everyone. This has been a really rich meeting, and I'm very happy to represent my colleagues, Matt Barnhart, Benny Kutiri, Lucine Gazarian, and other team members on our HIV vaccine team at USAID. It's probably well known that USAID works under the State Department in the United States, and Congress tells us what we can do through the foreign operations appropriations. And since 2001, we've been able to be working in the HIV vaccine space, so we're very committed to it. And we continue to get that appropriation, thankfully. And our partners have not really varied over the years. We've had IAVI as our prime partner doing HIV vaccine research, and we highly value IAVI as a partner. We also have AVAC as a prime partner in the advocacy, policy, and communication space. I would say that it's important to remember that USAID generally is not known for basic research and development, although in the contraception space, in the microbicide space, malaria vaccine, and HIV vaccine space, we have been investing for years. But these are anomalies, because at USAID, our mission is actually to build a world that is safer, healthier, and more prosperous for the people everywhere. So we invest in people. And I would say in my final remarks for the intro is that we really recognize the importance of traditional partners, including those that are based in the United States. But we really also are shifting towards non-traditional, underutilized local partners. And that is something that our administrator has given us marching orders to do. And of course, we're very happy to actually engage our local partners in the HIV vaccine R&D space. So that's all from me for now, Stacy. Thank you.
Thanks, Margaret. That was great. And I think lots to dig into there. So we will go next to Pervin Ankosaria, who is the Deputy Director of HIV and TB at the Gates Foundation.
Thank you. So I'm in the TB and HIV group within the foundation, and my role is to support and lead HIV vaccines and biologics development. And at the BMGF, we do have for HIV as an indication an end-to-end strategy, which is to accelerate the reduction of incidence. And we still believe that the vaccines will play an important role and a critical role for that purpose. We have, not us personally, but as a field, need to pivot to really go back and look at, while we've learned so much from our clinical studies of the past, we know that we continue to have this one of the key challenges, which is the requirement for an HIV vaccine to have a potent, durable, broad humoral and cellular responses, which we need to overcome viral diversity. And therefore, I think it's important as we move forward to test different vaccine concepts and combine those such that the effective vaccine can not only prevent infection, which is a very high bar overall, as we've seen even in the COVID situation, or at least stringently control viral replication at the site of entry and rapidly kill those infected cells to terminate that primary infection. And so the concepts that we are supporting and the field is excited about, as you can see from the work that has been presented and talked about even at this discussion, include the elicitation of cross-reactive potent neutralizing antibodies and CD8 T cell responses, either, as we say, as we've heard from Carrie, using the CMV platform, which sort of elicits a novel, not novel, but novel in the sense of MHCE responses, but also class 1 restricted responses, because in the end, we really do not understand what are the immune responses that we will need overall for a prophylactic and efficacious HIV vaccine. But I'm cautiously optimistic. I do want to end on that note, and I'm cautiously optimistic that based on our current knowledge of structural data, molecular dynamics, simulations, and all of the biochemical and biophysical information that we are getting, we now have a better understanding of how to precisely design immunogens, more than we've ever had in the past. We are at this inflection point. So I hope that doing these iterative studies, clinical studies, and establishing a robust preclinical pipeline will help us. And I would say that working towards a single regimen sooner rather than later that contains not only multiple immunogens, but can stimulate both a broad humoral and cellular response is going to be critical. For this particular introduction, I will stop there, and then we can add more as we talk about it in the discussion. Thank you.
Great. Thank you so much, Pervin. That was really clear. And so our final panelist is Roger Legrand. Roger Legrand is the head of immunovirology at the CEA in France. The Alternative Energies and Atomic Energy Commission works closely, I think, as people know, with the French ANRS. So important perspective. Over to you, Roger.
Thank you, Stacy. I have to inform the community that I'm not representing the funding organisms. I am using the funding for our own research, but I know how the funding is working in Europe and in France, so I can comment a bit on that. So the first thing is that we had so far 2 large consortia in Europe that develop HIV vaccines up to the phase 1, phase 2 clinical trials, which is one led in the UK by Robin Shattuck, and the other one was led by Yves Lévy at the Vaccine Research Institute in France, in collaboration with JP Pantaleo in Switzerland. So these 2 groups were working within the last framework of investment from Europe. And we are coming to the end of this 5 years program. As I was saying before, the difficulty we have in Europe is that we don't have a sustainable mechanism for funding long-term vaccine development in the HIV field. So you will have difficulties to find the HIV word in the new calls from Europe. So meaning that there is no continuity in the development. So that's something we can discuss. The other thing is that we had been able in France to continue developing HIV vaccines because we have an agency, the ANRS, which has invested in HIV field specifically. And we maintain some kind of investment, however, so far, which was not sufficient enough probably to go up to phase 3 trials, but this make kind of continuity. And what we need to maintain actually in this kind of limited resources capacity for research is to have a clear top-down investment for the research. So INRS did that for many years and had a real top-down development program, and we need to advocate to continue that in the future. These need probably the fact that we need to design the roadmap. So I said by Bill Snow, I said before, I understand what Bill Snow was saying, that is difficult and could be kind of overwhelming our capacity, but I think for advocacy, this roadmap could be important in our field. So that's what we want to say. And the other challenge we have is that we are facing the fact that young brains, so young investigators, are distracted from HIV research for new attractive, including vaccine area, new attractive programs. So we need to make it attractive by investing more in these young investigators. Thank you.
Great, thank you, Roger. Really, really insightful thoughts. So we're can open it up for discussion and would really encourage people, if you have questions for this group or questions at all, I think to put them in the chat, or please raise your hand and come off mute and voice them audibly. We're happy for participation. I can open it up. I think that, I think that there's been such a rich discussion on this call so far about this sort of need for global collaboration, coordination, and sort of a potentially a new strategic plan for vaccine, vaccine research or ultimately vaccine R&D. And one of the things that strikes me, I think we've all been in this field for a long time, and I think that there has always been this sort of emphasis on collaboration and coordination across the various entities that are funding and conducting the research. And I'd love to hear thoughts about, if we kind of make this new roadmap or make a new, HIV vaccine R&D strategy, what should we do differently? Let's just sort of dig into the nuts and bolts of what developing this sort of overall plan or roadmap might look like. I see Roger, you came off mute, or Carl, go ahead. Anyone? Yep, Roger, go ahead.
Yeah, I will rephrase a bit what I was saying earlier, but I think one thing we may need so far is to have a real agile continuum for R&D accessible to academics, so they can bring the product up to the production level and do the clinical trial with this capacity. So, and for this, we need a kind of coordination of this continuum and make it agile through facilitating the contracts and facilitating going from the preclinical to the clinical trials. And that's a very important thing. And I think could be a task probably for the HIV enterprise.
Okay, thanks for that. Pervin, go ahead.
I would say that without getting into the weeds, it will be critical to engage scientists from across the globe, particularly in communities where the vaccine will have the most impact. And we should really think about developing a vaccine with the end user central to that vaccine development activity. We need to have practical solutions for countries where this impact of the vaccine will be the greatest. And so I would urge our community to include scientists from across the globe and not just, people that we know all the time. We need new ideas, and we need new approaches for this development. And over.
Great, thanks, Pervin. I think that's so important, and obviously links, I think, to what Margaret brought up in her intro comments. Carl, over to you.
So I'm just going to echo what Pervin just said, and change it a little bit. Is that great ideas know country borders, and I think that what there is a need for is innovation, new approaches and new ideas, and be willing to take some risk. And so for that, we have designed programs where, from the beginning, no new data was required. And we all need to really look at the ideas coming forward and be willing to step outside our comfort zone and support these truly novel concepts. So to me, it's about being able to, through the mechanisms that we each have within our oversight, accomplish that kind of a goal. The other comment that Roger made about the production of material, being able to take something that looks promising and put it into GMP is expensive and a challenge, and it's something that the NIH has been willing to tackle and will continue to do so. It's complicated, it's difficult, but we have been able to help investigators make their vaccines. And third is the importance of ongoing dialogue and partnership with industry. We have had investigators like Dan Baruch that pushed forward with Ad26, found a very strong partnership with Janssen that the NIH and the Gates Foundation were able to get behind and really support. I think that model needs to continue to be used, and let's just see where we can go. But let's focus on innovation, and let's leave some of the ideas that have not delivered behind, and be willing to part with those.
Great, Carl. Thank you. Others want to weigh in on that?
Yeah, I guess from the industry perspective, it's always, you know, risk reward, right? As you think about moving forward with different programs, and so you want things in different buckets, right? You have the high risk, high reward bucket. You have one kind of in the middle where the risk is kind of middling, but you know you have a good probability of success, and then you know the really safe ones. And so I think we do kind of need to have, you know, things in each bucket, but be able to really explore those and have the capability of doing that. So that's one way to think about things. And you know, getting back to the whole manufacturing piece, that's a constant challenge, even for those in industry. I mean, the capacity worldwide, as well as the raw materials to be able to, you know, generate the amount of material that you need for clinical trials is always a challenge, and it always takes, you know, you have to reserve spots months or years ahead of time to try to do that. So I think one potential way is, you know, like Carl was saying, you know, investing, you know, or having some organizations invest in some of these earlier or smaller kind of manufacturing plants that can get things at least into phase 1, you know, faster.
Okay, good. Thanks for that. And Margaret, I want to go to you because I did see you come off mute, and then Pervin will come back to you.
Thanks, Stacy. Again, such a rich discussion, so many important points, and nice to hear that the NIH is really pushing the envelope when it comes to who's doing some of the research and bringing in innovation on many levels. I've been here for a while, as many other people on this call, and it strikes me that the pronoun we has not been as inclusive as it needs to be. And I'm not trying to be politically correct here. I'm saying that when you listen to Pervin and others on this call talk about the challenge scientifically, I've always been plugged into the biological and scientific challenges of finding an HIV vaccine for the world, but the political ones are no small matter. And I'm really referring to everything from having domestic resources added to the pool of resources from each country where HIV remains an epidemic, and to have the we include our African and other LMIC scientists in a very real way. I think we have not always done as well as we can to bring our colleagues to the table and make sure that their ideas are heard, and really open a space for our colleagues in LMICs who have been working very hard at testing these products to help us design them. I know that's a tall order and it might sound rather dreamy, but something Linda Gale said earlier too about, you know, first in human trials really struck me. I obviously been doing first in human trials in Africa very successfully for years. And unless we push the envelope in all of these areas, nothing will change. So I'm not saying anything that is particularly startling here, but I do have to ask the question, if not now, when it comes to more inclusion in the scientific decision-making and leadership in our field. And so that's the place we're going to be pulling some of our LMIC colleagues, our counterparts, into at USAID.
Thanks for that, Margaret. And exciting to know that USAID is really, you know, putting their money where their mouth is with all of that and making that shift. So we're excited to see how that unfolds over the next several years. So thanks, and definitely would love to pick your brain more about that. But Pervin, I want to go over to you. You still do want to input, yeah?
Sure. I wanted to first of all endorse what Margaret has said and really say we are behind that sort of action plan, if you will. It's really important. I've said this before, we need community input and we need user-centered, and the users are going to be not across the globe, not just in high-income countries. So we really do need to. So thank you, Margaret, for making that point again and emphasizing it. The point I wanted to make earlier was with industry and having these connections. It's great. We have stood up, as Carrie mentioned, VIR before, and we work with industry across the board for many things. But what is really important from my perspective, and I think our community perspective at large, is to ensure that the products that come out of these industry collaborations are available as rapidly as possible in low-income countries as within a few months, if not immediately, once a regulatory approval is had. And I think that is critical because people have contributed and communities have contributed to the development of these vaccines and or any other products for that matter, and then not having them available is a shame. We saw that in COVID, and we see this across many different product lines. So I think it's critical that whatever collaborations we initiate, that piece is built into that collaboration, ensuring that access is there right away. Thank you.
Thanks so much, Pervin. And I think it's such a critical piece. And actually, this is sort of running through my mind as the panelists were giving, everyone was sort of giving their overview. And I know that this issue of industry engagement is so critical, and we've seen this sort of a bit of a roller coaster over the course of HIV vaccine R&D history. I think especially with mRNA, right there, there's this sort of mRNA and I think the promise of mRNA helping us kind of iterate in this field and really kind of get to the bottom of the scientific hurdles. And I wondered if we could maybe dig into that a little bit more, Pervin. What steps, and I know this came up earlier in the conversation around kind of IP issues. Carrie, you talked about the lawyers that get in the way. But I think the experience of COVID has been, especially with Moderna, but to also to some degree BioNTech, has been so telling around just sort of the practically how this will play out and what can, especially with this sort of dependency on mRNA technology and biotech, pharma industry, Moderna, what steps can we put in place now? And I think I also just want to emphasize that this is a question that is coming out, I think globally in the advocacy community that is really raising some flags around HIV vaccine development. So how can we put steps in place, and then how can we communicate what's happening now around kind of industry partnership to make sure that we do have fast, equitable global access?
Maybe I can just speak quickly. From our perspective, we build in the staged approach to global access. So it's very difficult to ask for everything when we don't even know what the product is, right? But as we march further and further up, we have agreements in place, but those agreements introduce different elements as we get closer to product approval, for example, and we start building those in early, and we start talking about those with industry early, such as pricing, which countries we build in very early, which countries we think the global access is going to be critical. But the pricing and what the components are can come later once we have an idea as to what the product is. So I would say that we build those into our initial agreements and investments. Over.
Excellent, thanks. Carl, I see you're not on mute. Did you want to weigh in, or
no,
anyone else?
Not on this topic.
Stacey, can I just insert something very short here? USAID only invests in products that we think will be accessible and affordable. Otherwise, it's really not the business of a development agency to be trying to develop products that aren't going to be appropriate for LMICs. So full stop there.
Okay, thanks. Maybe while we're on this this topic, there's some conversation in the chat about kind of de-risking platforms, and I wondered, Carrie, would you be able to sort of weigh in on the question, how do we de-risk platforms, and when would a platform be considered as de-risked?
Yeah, so I think it's a complicated question because, you know, a platform can work for one disease but may not work for a different disease, and then you get into the details. For example, you know, what we talked about earlier in some conversations, you know, if you make some changes to an antibody, that can be considered a new drug even though you're using the same platform, and then you have to kind of repeat the talks and some of the phase 1 studies. But you know, for example, the mRNA platform obviously is a platform that worked very well for COVID, and we can use those tools to iterate on the HIV vaccine side. I mean, the question is, are you going to be able to generate enough, you know, of the antibody or product to be able to prevent infection? That's a whole other question. But at least you can utilize some of that technology to move, use that technology to move the field forward from that perspective. I mean, I think from our own, you know, for the CMV platform that we have, you know, we're in the clinic first with HIV, but, you know, if we're able to elicit these, you know, specific type of T cell responses, then there could potentially be, you know, moving into TB or some other infectious diseases as well. So once we get a proof of concept in one disease, you know, there is a potential of moving that into other disease areas as well. And then similar, you know, for our antibody technology, obviously their first one was COVID, but we also have an antibody directed against hepatitis B surface antigen, and we also have one directed against flu. So they may work slightly differently in the diseases, but at least the platform and what we have in terms of like the FC domain and those changes, you know, we hope can, you know, perform across different disease areas. So I think it gets into the details. So I think each situation is going to be different when you look at platforms and how that can be applied.
Okay. Thanks for that. Carl, over to you.
No, I just want to come back to this issue about platforms versus other strategies. I think the fundamental issue we face is the lack and the need for more innovation. We need novel methods, novel ideas that then can go into platforms so that they can be quickly iterated. We can sit here and talk about how great messenger RNA is till we're blue in the face, but there's an expression, what you put in is what you'll get out. You can fill in whatever additional noun you wish to put into that sentence. But to my mind, we really need to think specifically in the space of innovation. What are we going to do differently? We've seen 702, 705, and other vaccine studies show little or no efficacy, going all the way back to RV144, and then the follow-up, little or no efficacy. We really need to take a step back, and that's something that the NIH has been doing, and see what else is there. And one of the things we have that remains innovative is what Carrie's been talking about and Pervin talked about, is the VIR approach using the CMV vector. That remains a really interesting strategy, and it needs iteration. Nobody's going to nobody's going to get their first pancake perfect, but we really cannot just say, oh, it didn't work and move on. We really need to understand why things don't work and what can be done to either tweak, improve, or modify in a way that'll bring this field along. Also, from my perspective, we need to focus on innovation, and it will be great when we get to a point where we have a vaccine where we can discuss pricing. But I think it's easy to talk about pricing, and that's one of the reasons we all engage in that. But let's spend more time thinking about innovation and pushing forward on the science rather than worrying about something that's maybe 10 or 12 years off, if we're lucky. I hate to be a curmudgeon about this, but I'm trying to create a sense of urgency for ideas so that we have a robust, innovative pipeline. Thanks.
I think those are really, really great, great inputs, Carl. Did anyone want to follow on that? Maybe Carl, it's a nice segue. I think that the question that came up from Roger in the chat about, and obviously this is a little bit selfish, but I would love to hear the thoughts from all of you about if there's a need to reimagine the advocacy messaging for an HIV vaccine. And something that we talked about in our small group a bit is is this sort of sense of urgency, right? I mean, we've been at this for almost 4 decades, and now we're sort of at a bit of a pivot in the field. And so, how do we say, hey, we've been doing this, but it's still urgent, and this is why we need to kind of take this different approach. So, is there some sort of a kind of a big picture message that we can put out there? And I'm thinking, broadly across stakeholders, right? I think that the HIV Vaccine Enterprise, lowercase, has had strong support for since the beginning across the board, from kind of the funding level all the way down to the communities and the trial participants. But it feels like we might be in a space where we need to kind of retell the story, and certainly I think get the message straight on why we're kind of taking this pivot right now, and why there is still really this urgent need around an HIV vaccine. So I don't know if anyone wants to jump in on that, or Roger, did you want to add to that?
Yes, thank you. And I think it's very important, because I've been in a number of forums in the past few months, and sharing with you my personal opinion in there is that the way we talked about before is very much working in a bubble. And outside that bubble, HIV is no longer perceived as a problem. Even we heard in a recent meeting that, even in African countries, governments were not that much interested in getting support for health issues. They wanted funding for other issues. So there is clearly, in my view, a disconnect between our bubble, where we think it's important, and outside. And that's also an opportunity to bring back the community. And I always make a reference to the fight for PrEP in the UK, where I used to live, when we had to go outside the Ministry of Health and bang pots and pan outside, and that it took 10 years in the UK to get PrEP on the NHS. There was a real movement. There was an advocacy movement. And we don't have that to the same level. We have conferences where we talk to each other. And as Yves Levy said in the meeting, a lot of what we said has been said before. And for me, I would really like to find out, how do we move forward? How do we bring back HIV vaccine in the conversation? COVID has brought vaccine back into the public sphere. People were interested. People knew everything suddenly about vaccine. But the only one who were wondering, okay, so what does that mean for HIV? We're the one who were already working on HIV vaccine. And I'm just struggling with, how do we not make more noise, but how to reimagine the message? Because people will tell you, well, it's not a problem. There is treatment, there is PrEP, there is this, there is that. And we heard from Maureen, that's not going to solve the problem. And we heard 2016, I believe, Kevin Decore saying, we won't treat ourselves out of this epidemic. And this message, has disappeared. I'm stopping here and give way.
Thanks, Roger. Francois?
Yeah, in fact, I'm going to expand to what Roger just said, because it's a very lively and very productive meeting today, but one of the many of the closed bubble type of meetings. And the fact what is missing today is the link with the big picture of what's happening in the vaccine area. And perhaps WHO is trying to put all players in the vaccine field in line. And I'm just referring to what they produced like a few weeks ago, called the Global Vaccine Market Report, where they really try to think globally on how to prepare with vaccines to the next pandemics. And perhaps there is a strong need for HIV research community and those around this table to join this effort and not consider HIV vaccine research as a separate field at this stage. So I'm struck that the word WHO and WHO among other international organizations is not even listed in a discussion like today.
That's a great point. Thank you. Roger, over to you.
Yes, it's in the same kind of discussion actually that we, at least from the European perspective, we should not disconnect HIV from the other application of vaccine development in other diseases. So I think this is very important for advocacy. And we need to go beyond the impact on public health. We can also advocate in the fact that investment in this kind of challenging innovation, investment in capacity for doing clinical trials, investment in small plant for production of the vaccines, is also investment in industrialization of Europe and creating an economic ecosystem that can be developed then by itself. So we need to try to see this kind of idea that is not only public health, is covered also of investment economics and of investment in our sovereignty to do our development for the future.
Great, thank you for that. Before we sort of, I'm going to kind of wrap up with one final question for all of the panelists, but before we did that, I did just want to take note, and Margaret, kind of go back to you, because someone did raise in the chat, I think this issue you brought up around kind of including researchers across the globe, and especially from LMIC. And I wondered if you could you sort of provide, a little more texture from the USAID perspective, and certainly kind of the shift in USAID around this kind of leadership from the Global South, and what that might look like in the real world.
Yeah, I actually, thanks, Stacey. I saw that comment in the chat. I have to say that, we have long had partners, this includes the NIH and the Gates Foundation. We've long had partners who have track records, and we choose them on purpose. It makes rational sense. USAID feels that it's time to take our own risks with regard to directly funding local partners. So in fact, this morning we put out a new offer for our HIV vaccine R&D that is eligible only for African partners. And there is no, we're not actually thinking that we have to throw the baby out with the bathwater. We need the West. We need our traditional partners. But we have to reach across to our LMIC scientific partners in a more genuine way, so that they are in the seat of making some of the decisions and leading some of the discussions. And that actually hasn't happened to date. So that's the space we're in right now, is taking that chance and saying, what can our African partners do for themselves on their own with our help? Appropriate help. Yeah. And then, Stacey, may I just make one comment about the urgency question? I wonder if there's a fire that's bigger than the one you started years ago, everyone's going to focus on the bigger fire. The urgency matter is, I think, needing to be revisited, including, PEPFAR, the President's Emergency Fund for AIDS Relief. HIV is still a problem very seriously, especially in the more recalcitrant, hard to reach populations. We all know who they are. And it just like in malaria and other diseases, unless we reach those harder and very hard to reach populations, we will find a resurgence of the epidemic. So having said that, I wonder if urgency is the wrong word and essential, or something like essential, would be the right word. Tony Fauci always talks about the toolkit. Well, the toolkit in 5 or 6 years is going to have much more effective tools against HIV. The question is, where does a vaccine fit in? And we could just talk about the choice agenda. Right now, if we had freezers filled with an effective HIV vaccine, many people would choose to take something else. And we have to keep that, I think, in perspective. A vaccine will fit into an established set of tools for biomedical prevention of HIV vaccine or HIV prevention. And we must continue the pursuit of it, because it is potentially the most effective, cost-effective, and efficient tool that doesn't require taking pills, et cetera, et cetera. So I'll leave it at that, but I do wonder if the word urgency is no longer as relevant as essential.
Thanks, Margaret. That's so well said. And I think a call to action for us in the advocacy space. And also I think just, I think there has been very much a shift of kind of the de-siloing over the years, but I think it really is, I mean, especially as we're kind of all thinking about like the advent of injectable CAB and these sort of, better, longer acting products, how do we sort of increase that decrease the silos that do tend to exist, I think, in this prevention space still. So that's great. I'm going to ask for one last round robin from each of our panelists, and the question for you is just a little provocative, maybe fun. I mean, all else aside, in the positions that you each hold, what would be your big bet over the next, and some of you have spoken to this, Carl, I think we heard some great thoughts from you about this, but just kind of digging into it a little bit more. What would be your big bet to really move the vaccine field forward over the next 5 to 10 years? It just maybe in 1 or 2 sentences to make it easy.
Sure. My big bet will be on structure-based design of immunogens. Everything we tend to do with the GP120 or stabilized forms fails at some level. So we really need to follow Bill Schief and his strategy where we design very specific, very targeted immunogens that precisely engage the B cells and then precisely mature the B cells and precisely move this along. Because the vaccines we build must be better than what natural immunity can provide. And Bill is the first pioneer to give us such a product that is better than the initial immunogen. So to my mind, the trail has been lit. It's just a matter for others now to pick it up and follow that lead, because each target changes for each of the broad neutralizing antibody binding sites. I'll stop there.
Thanks, Carl. So clear. I'm going to go in alphabetical order again. So Carrie, I'll go to you next.
Yeah, no, I think we've learned a lot from the COVID field in terms of how to accelerate some of the trials and platform trials and support. So I think if we can get to some point like that in the future on the HIV side, because everybody's doing their own thing. Well, I mean, HVTN is sponsoring a lot of these different trials, but how can we integrate and more efficiently evaluate these different strategies going forward? I think many have talked about B cell and T cell strategies, and likely if we combine the two together, we can make better progress. So I think trying to figure out how best to like platform trials or other things to be able to facilitate these evaluations would be very helpful.
Excellent. Thanks a lot. Margaret?
I'm smiling because in the big bet arena, Carl has the most money. So of course we should just go along with Carl on this one. But we have a couple of dimes too at USAID, and of course we believe in the germline targeting approach. But I have to say that we are very consistent about our big bet being on people. So in this case, what we really want to see is that as the various innovative approaches to a safe and effective HIV vaccine continue to emerge, because we believe they will, we want the right people at the table. And not just because it's the right thing to do morally, but I think we need to really look at, as the field is more complicated, who's going to step up to take these vaccines? And if their own colleagues, friends, family, people that they meet at the market are not involved in the development, it's going to be a very tall order to ask them to be involved in the studies. So we need to see the communities be more inclusive all the way around, including the scientific community. So that's all. Thanks, Stacy.
Thanks, Margaret. And I think we've learned a lot from that in the COVID experience, obviously. So, Pervin, over to you.
The big bets, I would say, are from a technology perspective. I really do think we need to understand immune responses really well to know what a real efficacious vaccine would be. So I would say that in the context of HIV, to get all of the different arms of the immune system working coordinately and comprehensively to really protect ourselves, because what we really want is prevention of acquisition, which is very, very difficult. So that would be the big bet. And I would say that bringing people along and education, as well as strengthening regulatory and clinical services and laboratory services globally is going to be critical, because we are not going to be able to do this in one region only. And Margaret put it really well. We need to make sure that we bring the community together. Thank you.
Great, wonderful, Pervin. And finally, Roger, you have the last word.
Yeah, my last word in the continuation of Pervin comments is that we need to bring young people with good brains in order to make long-term programs they can take in charge of for the future vaccine development. So many of the communities getting old actually around the scientists, and we need to make that attractive so that these new investigators can put a lot of effort in the development of these very challenging vaccines.
Excellent. Well, I think that wraps us up. But just really to say, I think these are such brilliant and really critical perspectives from each of you. So thank you for bringing the succinctness and the frankness to the conversation. I think it's a really nice cap off to the session. So, and I believe I'm handing it back over to Susan.
Yes, you are. Thank you so much, Stacy, and thank you to all of the panelists. And it's really to me to thank everyone who's participated today in these sessions. It's been a very rich conversation. We've traveled from what the products should be and how we decide what to move forward, who's at the table, where and how the trials are done, how to engage regulators, and how to truly engage with community. And so we've really covered a lot of different issues. There are notes that have been taken, the meeting's been recorded, and a summary statement will be circulated after the meeting. Just again, a big thank you to everyone who participated and led different sessions and participated in different sessions and gave us your thoughts and opinions. It was really a very rich discussion, and we want to thank everyone. So wishing you the best for the rest of the day, and thanks again for participating.